the characteristic of the soil is they have low organic carbon content, high soil acidity, low pH, low nutrient tension, low soil CEC, poor microbial activity, and then also poor soil structure. Biochar can improve those poor properties of acid soil. The incorporation of biochar will increase soil carbon sequestration and also can act as liming agent, improve nutrient supply, increase soil pH, and then lower soil acidity, and then also promote microbial activity, and also make a better soil structure. We can apply biochar in soil. It's not only in form of biochar, but we can also make co-compost biochar by mix biochar and compost, and then continue the process in composting manner, and the product is co-compost biochar. We hope that co-compost biochar will promote better result than biochar only. The aim of the study is to measure the effectiveness of co-compost biochar to reduce soil acidity and to improve soil chemical properties, also thus increasing crop yield. The location of the study is in Taman Bogor Research Station in East Lampung. The first season is started from December 2017 up to April 2018, and the second season is from April to July 2018. The soil type is typic Kanha with sandy clay loam texture class. The experimental design is randomized complete block design with 4 replication. The treatment is control, no ameliorant at all, and then biochar cacao shell biochar 10,000 per hectare, corn cob biochar 10,000 per hectare, and cacao shell co-compost biochar, co-compost corn cob 10 per hectare, and manure 10 ton per hectare. This scale 4 to 1 is the meaning is that biochar is 4 and 1 is for cow dung manure, as the same as manure here. And the fertilizer is 300 kilogram per hectare of urea and 200 kilogram per hectare of NPK. The plot size was 15 meters square, and the crop indicator is maize BC18. And this is the table is show about the initial soil chemical properties, which the pH is acidic 5.33, and all the base cation, calcium, magnesium, and potassium is also low. The CEC is low, 8.9, and the exchangeable aluminum is high, 1.59. So this is the chemical ameliorant properties of the ameliorant that used in the study. The best quality of ameliorant is belong to cacao shell biochar. So all the parameter is the highest compare other ameliorant. So the pH is alkaline and the highest pH is belong to cacosel biochar, and the lowest is belong to manure. And this is the soil chemical properties of after the application for planting season 1 and planting season 2. And we can see here from the red shape here that the effect of soil ameliorant on pH, the best performance is on cacosel biochar 10 ton per hectare. Compare other the ameliorant. We can see from the table that cacosel biochar is significantly increase soil pH. Meanwhile, other treatments are no different from the control. It's also the same from soil organic carbon, nitrogen, and potential phosphorus and potential potassium, and also for available phosphorus. Meanwhile, on the second planting season, we can see that the effect is still on the treatment when we talk about soil pH, and it's again cacosel biochar 10 ton per hectare is the best performance on improving soil pH. Meanwhile, others like corn cob biochar, co-compost biochar, and manure is no different compared to control. And other effect on total nitrogen, there is no significant difference among treatments compared to control. Also for potential, potential phosphorus, all the treatment have the same effect, compared to control, meaning that the biochar treatment, the all soil amendment treatment has effect to increase potential phosphorus compared to control. And this is about calcium and magnesium. During 2 planting season, all the treatment were increased soil calcium and magnesium content. Can see from the picture, from the graph, that from the first planting season, that the cacosel biochar treatment 10,000 per hectare is the best performance also to improve calcium content in soil. And from magnesium, that the same effect also from for magnesium, that cacosel biochar treatment has the best effect on improving magnesium content in soil. This is about aluminum content after the treatments. So all the treatment effectively decrease aluminum content in the soil. And from the shape here, that we know that also the best performance is by biochar cacosel in the first planting season. In the first planting season, aluminum content is can say that is zero in first planting season due to cacao shell biochar treatment at 10 ton per hectare. And this is the significant negative correlation between aluminum content and dry grain. So we can see that when the aluminum content is higher than 2 centimole per kilogram, then the dry grain is very low also too. But when the aluminum content is in the range of 0 to 0.5, then the grain is in higher position. And this is the graph of the dry grain and dry biomass for planting season 1 and planting season 2. And we can say that in the planting season 1, all the treatment have good effect on increasing dry grain compared to control. Conclusion, the co-compost cacao shell biochar and corn cob 10 ton per hectare dose for 2 planting season increase soil pH, total nitrogen, potassium, calcium, and decrease exchangeable aluminum. The application of cacao shell biochar resulted in a more significant improvement of soil chemical properties. The dry grain from the biochar and co-compost biochar application increased significantly compared to control, but cacao shell biochar and co-compost cacao shell biochar gave more stable result. During 2 planting season, maize dry grain increases were correlated to aluminum. Good quality cacao shell biochar has more significant meaning when used not to as co-compost. In contrast, corn cob biochar, lower quality should be formulated as co-compost. The amelioran, which has biochar as the main material, either with or without compost mixture, could improve soil chemical properties and increase plant productivity in acidic upland and be applied only in the first planting season with no additional application in the second season. Thank you for all your attention.
Thank you, everyone. Thank you, everyone. I am presentation my research, a title of okay, I'm sorry. I'm presentation my research, a title of efficacy of milk moringa leaf extract and co-manure to soybean growth and yield. My name is Haryuni. My team Isnan Yuda Utama Atmaja, Teguh Supriyati, Sabto Priyati, Bagus Budiono, Rahayu Rumaningsih. We come from Department of Agriculture, Universitas Tunas Pembangunan, Surakarta. Background my research, soybean is an important commodity in Indonesia that acts as a source of vegetable protein used for tempeh, tofu, oil, cakes, and beverage as well as other forms. In 2018, the harvested area was 680,373. Soybean productivity was 1444 quintal per hectare, and average recruitment was 3 million tons per year. Soybean imports in 2020 reached 1 million tons from soybeans producing countries. The Grobogan variety of soybeans has an average yield of 2.77 tons per hectare and a potential yield of 3.40 tons per hectare with a weight of 100 seeds of 18 gram. Moringa leaf extract functions as a biofertilizer because moringa leaf contains crude protein, crude fiber, crude fat, as moisture, carbohydrate, and calories. Co-manure has the advantage of increasing soil humus content, stimulating soil microorganism, and improving soil structure so as to increase the availability of nutrient for plants. Co-manure is given to accelerate the availability and utilization of nutrient for soybean, whereas co-manure contain macronutrients such as nitrogen, phosphorus, potassium, magnesium, organic carbon, CN ratio, and other ingredients, organic with water content. Purpose of the research to determine the effect of the dose of moringa leaf extract and co-manure on the growth and yield of soybean. Research methodology. Moringa leaf extract dose variation, where 0, 20, 0, 10, 20, 30, 40, 50. And every 2 weeks up to 4 times. The second factor with the dose which is 30 and 60 gram per polybag, added every 2 weeks up to 4 times. My research at Jakalan Village, Karangnongko District, Klaten Regency, Central Java, from October to December 2020. The altitude is around 300 until 400 meters above sea level. The soil types was Recosoil. Result of the analysis showed a significant difference. Conducted a further test using Duncan multiple ring test at the level of significant difference 5%. Result of the observation. Table 1. Effect of moringa leaf extract to height, tray white, number of pod, and weight of seed per plant soybean. We can see table 1, all parameters significant different. So, table 2, effect co-manure to height, tray white, number of pods, and weight of seed per plant soybean. We can see only plant height parameters significant different. The others parameter not significant different. So, table 3, effect of combination of moringa, leaf extract, and co-minor. Only plant height parameters significant different. So, and the other parameter not significant different. Conclusion. Effect of application of moringa leaf extract and cow manure on soybean, the result shows that the combination treatment of M5P2 at a dose of 50 milli per liter of moringa leaf extract and 60 gram per polybag of cow manure had the highest growth and yield compared to other treatment. The benefit of this research is to provide information on the use of appropriate doses of moringa leaf extract and co-manure on soybean growth and yields. Thank you for your attention.
All right. Thank you very much for all of the speakers, Mr. Juli, Mrs. Jubaida, and also Mrs. Haryuni for giving such as comprehensive information and presentation for our first sections. Okay, ladies and gentlemen, now we move to our discussion time. We will have 6 minutes. So here we have some question in the chat box from Mrs. Rahma. Hello, Mrs. Rahma. She comes from Green, of course, with ID number 37. The question is suitable for Mrs. Jubaida. And the question is the co-compost biochar easy to apply by the farmers? And is the compost biochar has economical value? Please, Mrs. Jubaida, to answer Mrs. Rahma's questions.
Okay, thank you, Mbak Rahma, for your question. So you asked about, is it easy to apply by farmers? Apply in terms of application is, I think it's not, I think it's easy, but in terms of biochar production, then it's another question. I mean, it's not really need a big labor to apply biochar in the field, but when you produce it, it's a lot of work. It's laborious, and so in our experience, that farmer has hesitation to apply this technology, to adopt this technology in terms of production. They do not deny the, let's say, the effect. They agree that biochar is contribute to the improvement of their result, but when they talk about the production, then it's not easy for them. So is it easy to apply? Yes, it's easy to apply, but it's not easy to produce. And the second question is the compost biochar has economical value? Yes, it is. Okay, maybe then when we apply more, not apply, when we calculate more, is it economical value? Maybe we should see how the capital to produce biochar. It's biochar. We suggest that biochar use in situ organic matter, so that you do not need any additional cost to send biochar from other region to your place. So maybe when it's in situ and you can provide the labor cost, then it's economical. It's economical. They can earn some economical value in terms of that. But then when you need to import, let's say not import, you need some additional money to send the organic matter to in situ or not from other place. I mean, when you need the feedstock from other place, then maybe it's not economical. So I think that's my answer for Mbak Rahma question. Thank you.
All right, thank you, Mrs. Jubaida. Or maybe there is some feedback from Mrs. Rahma to Mrs. Jubaida. Nothing? Okay.
Thank you
So
very
it is
much.
enough. All right, you're welcome. It is enough clear, I think, because the important thing that Mrs. Jubaida said is about the farmer had curiosity to learn how to farm, how to produce, and the etc. All right. So actually, there is one question more, but from Mrs. Jubaida, from Brin also to Mrs. Haryuni. The question is, why did you use extract moringa as biofertilizer? Why not moringa compost? Thank you. Please, Mrs. Haryuni.
Thank you, Miss Zubaida. Because I making with my student and making it easier and faster. And besides that, it also does not require mixed ingredients. Only moringa leaf. I use only moringa leaf without the other apa ya? The other component,
Materials.
Yes, the other component without the other component. Okay, I think it. Thank you, Miss Zubaida, Mrs. Zubaida.
Okay.
Are you?
Okay.
I need to elaborate more, I think. So for farmers, it is common for them to use compost. Then it's also that they can make compost by themselves. When you use moringa extracted as biofertilizer, is it? I don't think it's easy for farmers to do that by themselves.
Yes, I used to student not farmers, because I used to student not farmers, and only area not inland.
I see.
I have not land. I have not land, or I have not area to making compost. I have not. Yes.
So it's some kind of initial experiment, I think. Okay, thank you, Ibu.
Thank you your attention. Thank you.
Thank you, Mrs. Haryuni and Mrs. Jubaida. Mrs. Haryuni said that actually the problems is they have no area to make a compost in their university maybe, and then of course they have students that also want to make some moringa extract with easier and faster like that. So, okay, ladies and gentlemen, now we move to our second sections. Allow me to welcome to our next presenters. There will be presenters with ID number 72, Mrs. Sulfahmi, or Mr. Sulfahmi, I think. Hello, Mr. Sulfahmi. Are you here?
Yes.
All right, Mr. Sulfahmi. All right, and then second presenters, there will be with number ID 81, Mrs. Hidayatul Arisah and team, are you here in this room?
Yes, I am here. I will present.
All right. Yep. Thank you, Mrs. Farida. And then the last presenters, there will be presenters with ID number 82. There is Mrs. Juartina. Are you here, Mrs. Juartina?
Yes, I'm here. Ready
All
to
right. Ladies and gentlemen, with no further ado, we will come together, our operator, to play the video from our third presenter.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Thank you very much for this opportunity. The first, I want to introduce myself. I'm Zulfahmi from Agrotechnology Department, Faculty of Agriculture and Animal Science, State Islamic University Sultan Syarif Qasim Riau, Indonesia. I would like to present the research with the title Phenotype Performance of the M1 generation Bima salad. Allium cephal variety Ascolanicum, result of ethyl methasulfonate in usage. Salad is the second horticulture crop important in Indonesia after chili, and is used for culinary, spices, vegetable, and medicinal properties. In Rio Province, the salad of Bima variety has been cultivated by many farmers and has sufficient adaptation compared to other varieties, even though the production is still not yet optimal. According to the statistics of Rio Province 2021, the mean productivity of salad is around 4 tons per hectare. That's still far from the average national productivity is 9.5 tons per hectare. So, increasing productivity of salad must be implemented via improvement of cultivation practices and planting material. Ethyl methasulfonate is one of the chemical mutagens often used to induce mutation and expanding of crop genetic variability. EMS induced the point mutation, so that EMS is ideal for producing missense and nonsense mutation, which would provide a series of changing of functional mutation. Many studies has applied EMS to increase genetic variability of salad, as reported by Joshi et al. 2011, Bihara et al. 2012, and Shukla 2017. The objective of this research was to investigate the phenotypic performance of salad of Bima varieties. Result of ethyl methanesulfonate induced. Solid bulk of Bima was obtained from the local source. The experiment was arranging the following random complete block design with 4 treatments: control 100, 200, 300, and 400 ppm of ethyl methanesulfonate. The treated and the control bulb were planted on the well-prepared plot at the spacing of 20 by 20 centimeter between row and plant. The plot size was 1 meter long and 0.9 meter wide, consisting of 3 rows per plot. All the other management practices were uniformly applied to all plots. The observation parameter included plant height, number of leaves, number of tillers, bulb weight, The objective of this research was to investigate the phenotypic performance of salad of Bima varieties, result of ethyl methanesulfonate induced. Salad bulb of Bima was obtained from the local source. The experiment was arranging the following random complete block design with 4 treatments: control 100, 200, 300, and 400 ppm of ethyl methanesulfonate. The treated and the control bulb were planted on the well-prepared plot at the spacing of 20 by 20 centimeter between row and plant. The plot size was 1 meter long and 0.9 meter wide, consisting of 3 rows per plot. All the other management practices were uniformly applied to all plots. The observation parameter included plant height, number of leaves, number of tillers, bulb weight, bulb diameter, bulb press weight per clump, and bulb dry weight per clump. Data obtained were analyzed by ANOVA, and if the EMS treatment was significantly different, that will proceed to the MRT test. This is the result of the research. The analysis of variance showed that EMS treatment were different significantly for all parameters. We look in the picture. Plant height and the number of leaves of the control treatment have highest value compared to all EMS treatment, and it is significant different. The highest reduction for both parameters observed in 400 ppm EMS. The number of tillers. We observed the treatment of the 3 and 400 ppm EMS was significantly different with control. The highest reduction of the tiller number was 35%, displayed by 400 ppm EMS. For the character of bulb weight and bulb diameter, the control treatment also have highest value compared to all EMS treatment. Bulb diameter and bulb weight have higher reduction in the 400 ppm, with the 79 and 39% respectively. Bulb fresh weight per clump and bulb dry weight per clump of the control treatment also have highest value compared to all EMS treatment. The higher reduction of the bulk fresh white flocculum and bulk dry white flocculum was observed in 400 ppm, with 74% and 78% of reduction respectively. Increasing of the EMS concentration caused a significantly decrease in all parameters studied due to mitotic abnormality gradually increased with increasing concentration of mutagen. On the other hand, ionizing radiation normally induces chromosomal deletion and rearrangement. The conclusion of this study was the phenotype performance of the M1 generation of bimasalot are not better compared to the control treatment. We suggest to planting M2 generation to find the potential mutant in the future. Finally, I would like to thank the Institute of Research and Community Services of Islamic State University Sultan Syarif Qasim Riau for funding of this research. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, moderator. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. May I introduce myself? My name is Farida Yulianti. I am a researcher of National Research and Innovation of Republic of Indonesia. Now, I want to present our research with entitled Genetic Diversity of Strawberry, Fragaria ananasa, for early bright mutant induced by gamma rays irradiation as revealed by ISSR marker. The background of the research was the fact that early bright strawberry, one of the superior strawberry genotype, has been domesticated more than 15 years in Indonesia, especially at the highland altitude with low temperature like Garut and Ciwidey, Bandung. On the other hand, climate change causes global temperature increases. Therefore, the availability of a strawberry genotype that is tolerant to high temperature is important. We know that breeding programs of strawberry can be carried out through conventional breeding and induced mutation. Commonly, in vitro gamma rays mutagenesis causing the release of electrons and directly disrupting the chemical bonds of molecules when they pass through matter, and producing a reactive oxygen species or ROS that damages all components of a cell, DNA lesion, DNA single-strand breaks, and DNA double-strand breaks. Gamma rays mutagenesis also causing macromutation that produce small phenotypic effect. Therefore, we think that early selection by marker-assisted selection is important. The objective of the research was to identify the DNA polymorphism among the early bright strawberry mutants as revealed by ISSR marker. To achieve the research goal, we used 36 mutant genotype and one mother plant as plant samples. The DNA isolated from leaves using plant genomic DNA kit and then amplified using 15 ISSR primers, separated by electrophoresis gel agarose method, and visualized using gel documentation system. The presence of a brand counted as 1 and the absent one counted as 0. The data obtained used to calculate polymorphism rate, polymorphism information content or PIC, multiplex ratio or MR, effective multiplex ratio or EMR, and marker index or MI, and UPGMA dendrogram. Results of the research show that all of the primers generated reproducible band with high polymorphism. The number of bands for each primer varied between 9 to 17, giving a mean 11.93 pence per primers obtained. The percentage of polymorphism ranges from 75 to 100% with an average of 93.3%. The PIC value for each primers varied from 0.4 to 0.5. The value indicated that the ISSR primers used were uninformative markers. The effectiveness of each primers used was measured by the marker index or MI. The MI of the primers used ranged from 2.38 to 4.85. High MI value indicated the gamma rays irradiation had successfully induced high genetic variation in early bright strawberry mutant. Based on UPGMA clustering, the ASSR marker grouped the genotype into 5 major groups. Cluster 1 contained 2 genotypes, Cluster 2 contained 9 genotypes, Cluster 3 contained 22 genotypes, Cluster 4 contained 3 genotypes, and Cluster 5 contained only 1 genotype. The closest mutant genotype to the mother plants was genotype 70 with a similarity index 2.1%, while the most distant genotype was genotype 64 with a 64 with a similarity index of 71.69%. Conclusions. First, the ISSR marker use assures suitability in discriminating among 36 strawberry mutant genotype and the mother plants, revealing the genotype variability of each genotype. Second, the genetic diversity among strawberry mutant genotypes show that 20 gray gamma rays irradiation generated a sufficient number of induced mutations. That's all my presentations. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opportunity for me to present our research with the title Screening of Simple Sequence Repeats Primers from Mutated Indigo Verazoliniana Plants. My name is Juatina Idaroyani. I am from BRIN. The background of our research: DNA profiling of plants using molecular marker is a useful tool for the identification and characterization of superior plants in plant breeding programs. This tool can shorten the time for superior plant selection, especially in plants with long growth cycles. Molecular marker work by revealing variation or polymorphism of the DNA sequences between different individuals in the population. One of the most popular marker is simple sequence repeats marker or microsatellite. Why? Because show codominance, highly polymorphic, hypervariable, and it is distributed throughout the genome and has a higher mutation rate. The molecular marker can detect rapidly the substitution mutation or point mutation, reassortment, insertion and deletion, replication error, and DNA tandem repeats. Mutated plants need to identification of genetic variation using a method that are simple, accurate, reproducible, and show the polymorphic between mutated plants to shorten time of the selection process. To use molecular marker with precision method, screening of primers that use is the main factor to obtain the optimal results. Selection of the optimal primer annealing temperature will increase the efficiency and specificity of amplification to obtain the suitable primer use. For this research, we use 14 SSS primers from same genus of Indigofera zollingeriana that proved to amplify Indigofera pseudotinctoria. So the objective of the research was to screen 14 SSS primers to know the polymorphic DNA profiles of 12 mutated plants of Indigofera zollingeriana. Material used for this research were leaves from mutated plants of Indigofera zollingeriana with 6 different doses. Material preparation, mutated plants from Indigofera zollingeriana with dose 0, 100, 200, 300, 400, and 500 gray were planted in the land. After 12 weeks planted, leaves were put into plastic clips and labeling in every sample for next treatment. For genomic DNA isolation, DNA genome from 12 mutated plants were isolated using Wizard Genomic DNA Purification Kit. For screening SSS primer, we use for 14 SSS primers, 12 DNA genomes, and amplification with multiplex PCR with 2 PCR conditions. All product PCR then electrophoresis with 1 or 1.5% of gel agarose. For optimization of annealing temperature, the gradient temperature was set with 6 different temperatures based on the original temperature with the gradient degree at 0.3 degree Celsius. We used primer that amplified the DNA genome but had thin bands. So the result of genomic DNA isolation, 12 DNA genomes of mutated E. zollingeriana leaves were successfully isolated using this protocol. Concentration of DNA genomes range between 345.6 until 3318.6 nanogram per microliter, and purity range between 1.56 until 1.90. For screening primer, screening with 40 SSS primers show that not of all primers in temperature of annealing at 59 degree, 5 primers show polymorphic, 8 monomorphic, and R4 primer couldn't amplify the DNA genome. In temperature of annealing at 48 degree, 13 primers show polymorphic and R9 primer monomorphic. We assume that annealing temperature at 48 degrees show more polymorphic profile than at 59 degrees Celsius. DNA profiles of polymorphic bands show 1 until 10 bands, and the best primer was R3 primer with 1 until 10 bands with clearly bands, following by R8 primer with 1 until 8 bands, but with thin bands profiles. The thin bands show that temperature of annealing insufficient to attack the primer sequence to the DNA genome and still need to optimize the annealing temperature. For this screening, we obtain 5 primers that show polymorphic band at 59 degree Celsius and 13 primers at 48 degrees Celsius. For optimization of gradient temperature, we only use R5 into R5.2 DNA genome for treatments. The results show that every primer had specific TM for obtained clearly DNA band. After treatment for gradient temperature, the results show that 11 primers are suitable for use in further DNA profiling of another mutated Indigofera zollingeriana. The specific temperature that obtained from gradient temperature will be used as annealing temperature for the next PCR. So the conclusion of our research show that 12 DNA genomes of mutated indigo Verazolingeriana leaves were successfully isolated. 5 primers show polymorphic band at 59 degree and 13 primers at 48 degree. Treatment for gradient temperature, the results show that 11 primers are suitable for use in further DNA profiling. The specific temperature that obtained from gradient temperature will be used as annealing temperature for the next PCR. The authors would like to extend appreciation and thankful to Research Organization of Nuclear Technology, BRIN, for providing financial aids during this research. This is the part of our reference. So thank you for the attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. That was a very comprehensive presentation for all of you guys here, our three presentators. It is always interesting too for me to talk about strawberry because it is my favorite fruit. Okay, now it is time to have discussions with our speakers. And in the chat box, there are 2 questions. First question comes from Mrs. Aida with the number ID 81, and the question is: are each of the primer include forward and reserves? Okay, please, Mrs. Farida, to answer the questions.
Okay, thank you for the question. Thank you for Ibu Aida Khalida. I think ISSR primers is a dominant market that has only one strand primers. And then for Ms. Ibu Jubaida question, what is the superior plants? Is it the same as Bibit Unggul or VUB? I think it's a superior plants. It's a new variety of strawberry. Thank you.
All right. Or there is some feedbacks from our audience here, from Mrs. Aida or Mrs. Jubaida? Nothing? Okay. If there are no feedbacks, we will move to the next sections. There will be 3 presentators. Wait a minute. All right. Okay. We will have presenters with number ID 105 with Mr. Supriyono. Mr. Supriyono, are you here in this room?
Yes. Ahmad Taufik, sorry.
All right. All right, Mr. Ahmad Taufik, as the team, of course. And then the second presenters, there will be with the number ID 124.
Yes.
Sorry, 124. Mrs. Muji Rahayu.
Yeah,
all right.
I'm here.
And then the third speakers, there will be with the ID number 130. There will be Mr. Supriyono and team. Are you here,
Yes.
Mr. Supriyono?
Yes, I'm here.
All right, Mrs. Talita Syahda. Okay, so ladies and gentlemen, please welcome together for the operator to play the videos.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Ahmad Taufik. I am a student of the student program Faculty of Agriculture Universitas Maret Indonesia. And this research was also supported by Professor Dr. Enso Supriyono MS, Professor Dr. Enso Sulanjari MS, and Aprilia Ekonomi Masari SPMSC. I would like to present my research about the effect of potassium fertilizer doses of growth and pouch of red ginger, Juniper officinalis varuflu. An introduction, red ginger has a prospect for export the traditional medicine, food and beverage industry, and cooking spices in Indonesia. The many benefits of red ginger cause the increasing of market demand both regionally and global for industrial development. In order to meet market demand, it is necessary to increase ginger production with both organic and inorganic fertilization. Plants need potassium nutrient to meet their nutrient, such as HCl fertilizer. The role potassium in plants is actually very important because affects psychological process and strengthen the tissue unit in large quantity in the research of research method. This research was conducted from March to August 2021, located in Palang Hamlet, Wonorejo Village, Jatiosodisi, Karanganyar Regency, and Central Java, Indonesia. And then that is material and tool. And then the research was carried out using RGBD or randomized complete block design. One factor consisted of 4 levels of treatment and 6 repetitions in order to obtain 24 experimental units. The data analysis is in this research was analysis of variance and continued with the DMRT or Duncan multiple test level of 5%. And next is results and discussion. The first is condition of the land. The research is located at an altitude of 800 meters above sea level and tropical climates. Ginger plants can grow at an altitude of 0 to 1,500 MISL. The air temperature on the experimental field was between 25 degrees Celsius lowest and 33 degrees Celsius highest. Rainfall during the experiment amounted to 2,655 millimeters. And then the type of soil found in this research is littoral soil. The soil is a relatively young soil type, coarsely textured, so it tends to be nice. The texture of the littoral soil is raw and tends to be nice. The nature of the nest makes the holding power of water by the soil low, so that nutrients are difficult to bind to pollutes, so that they are easily leached. The eggs and then the red ginger grow. The eggs of red ginger plant reach about 43 to 48.47 centimeters and does not differ markedly between uncultivated K fertilized dose of 150 kilogram per hectare. Ginger has only reached a height can be said to be low compared to what other research have achieved by about 71 centimeters. Plant height below average is generally the result of bad transplantation. The number of leaves is negatively correlated with the height of the plant, meaning that the height of the plant increases, the number of leaves actually falls. This indicates that the stem internus are increasing in length. However, this is not because of growth disorder, because the height of the plants and the number of leaves between doses of K fertilizer do not differ markedly. In contrast to plant height, the number of ginger leaf in study was much lower than in other studies. With the dose of K fertilizer 15 and 30 tons per hectare, the number of leaves is 144.04 and 156 strands. And then the number of leaves is not significant because the dose of fertilizer is related to the number of tillers, also does not differ markedly. This relates to the size of the buds, which also has no noticeable effect on the number of tillers. Biomass is accumulated of photosynthesis at the source, is the leaf and stem. Biomass therefore describes the ability of the leaves as an organ of photosynthesis. In this study, the fact showed that the number of leaves was closely correlated with biomass is 0.71, and biomass with the number of tillers was very closely correlated, is 0.87. And then the ginger yield. The application of KCl fertilizer is only significant on the variable fresh weight of rhizome per plot and the dry weight of rhizome per plot. Potassium is used for carbohydrate metabolism, regulating water content, and activating enzyme to increase the translocation of carbon to the rhizome of red ginger. The fresh weight of rhizome will affect the weight of rhizome that have undergone drying, where the ginger, the fresh weight of the rhizome, the height of cell weight will also tends to be high and vice versa. If the planting media lack nutrient, it results in a small ginger rhizome size, so that nutrient and water ability are needed that are always present in the planting media, so that the growth, pace, and development of rhizome are optimal. And then the correlation test, the ginger and the number of leaves. The process of photosynthesis can take place optimally, and the result out in the form of photosynthetic will increase. Photosynthetic results will be translocated to our plant tissues. The more the wet weight of the plant increases and followed by the dry weight of the plant, a large volume of rhizome is affected at the time of growth of the ginger plant. Ginger that grows and adapts well to the new medium will produce a large and heavy rhizome. And then the conclusion of the research-based method is fertilizing KCl up to 150 kg per hectare has not increased the yield of red ginger. The yield of rhizome self-weight of the hectare is only 1,439.8 kg. That is very low. Thank you so much for all your attention during this presentation. I hope you have a great day. Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. I will present my paper about upland rice growth on giving of biochar and organic fertilizer. My team Muji Rahayu, Aprilia Ika Nurmalasari, and Kartika Aprilia. For introduction, upland rice is an important food crop commodity in Indonesia, especially in dryland area. Upland rice is one of the food crops that can be cultivated on dry land. In generally, dry land has low soil fertility. Soil condition with low nutrient and organic matter when used for agricultural activities will produce less than optimal yield. So it is necessary to add nutrient to the soil. The addition of biochar and organic fertilizer will have a positive effect. The application of biochar is one solution to improve the balance of nutrient in the soil by increasing soil fertility. The application of organic fertilizer can also have a direct effect on plant growth. Organic fertilizer that are widely used are fertilizer derived from animal manure that have been fermented and contain various nutrients. Manure contain macro and micro elements that are beneficial for plant growth. Some research results show varying results. Giving biochar to the soil is also able to increase growth and nutrient uptake in the plant. Goat manure has a higher nitrogen content where the N content can increase plant vegetative growth. The problem in the research, the nutritional content of type of biochar such as coconut shell, rice husk, and strobilanthes biochar, and organic fertilizer such as chicken manure, goat manure, and cow manure, and other organic fertilizer is very diverse, so that the response of upland rice to the application of various types of biochar and organic fertilizer is also different. Research objective to study the response of upland rice growth on giving of biochar and organic fertilizer type. Methodology, research design. The research used completely randomized block design, RCBD, consisting of two factors. First factor is type of biochar, consists of without biochar, coconut shell biochar, rice husk biochar, and strobilanthes biochar. The second factor is type of organic fertilizer, consists of without fertilizer, chicken manure, goat manure, and cow manure fertilizer. Its treatment combination repeat 3 replication. The observed variable were plant height, number of leaf, number of tiller, leaf area, fresh weight of biomass, and dry weight of biomass. Data were analyzed using analysis of variant and Duncan multiple rank test on 95% level. Result, first plant height based on analysis of variance so that the application of biochar and organic fertilizer type were not significant to height of upland rice. Figure 1 show that the highest plant height up to 10 weeks after planting in the treatment of goat manure. Second, number of leaf and number of tiller. The analysis data show that the application of biochar gave a significant effect on the leaf number and tiller number of upland rice at the age of 4 to 6 weeks after planting. The best treatment with the highest number of leaf and number of tiller was found in the treatment on giving coconut shell biochar. A third leaf area. The analysis data show that the application of biochar and organic fertilizer type were not significant on leaf area of upland rice at the age of 4 and 8 weeks after planting. The largest of leaf area on treatment was applied to good manure with area 28 centimeters square and 757.2 centimeters square. 4. Fresh weight and dry weight of biomass. The analysis of variance showed that the application of biochar and organic fertilizer were not significant on the fresh weight and dry weight of biomass of upland rice. Conclusion. Application of biochar and organic fertilizer type was not able to increase plant height, fresh weight of biomass, dry weight of biomass, and leaf area of upland rice. Second, application of biochar was able to increase the number of leaf and the number of tiller of upland rice at 4 until 6 weeks after planting. And the third, coconut shell biochar gave the highest of the leaf number and tiller number than rice husk biochar and strobilanthes biochar. That's all my presentation. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Talita Nursyada from 11 Maret University. First of all, I would like to thank you for being given the opportunity to be able to present the result of the research with title The Effect of Liquid Organic Fertilizer on Growth of Bulgur and Yield of Porang Amorphophallus Mareri Bloom. For the first part is introduction. Porang is a plant of the Araceae family that has a very poor prospect because it has a lot of nutritional content and benefit according to the Director General of the Ministry of Horticulture as of 2020 in January until September. Export porang reached 1,931 tons worth $31,427,394 in the form of chips, flour, tubers in the form of slices or not slices. Porang production must be increased to meet market demand. And the application of liquid organic fertilizer, LOF, is expected to increase crop production and maintain soil fertility because LOF contains macro and micronutrients, plant growth, regulatory organic compound, and microbes producing biocatalysts. LOF application with foliar spray is more effective because it's applied to the entire leaf surface and it can be absorbed by plant directly. The aim of research, number one, is to examine the effect of LOF application on bulbil growth and porang yield, and the second one is to get the optimum LOF dose that has a better effect on bulbil growth and porang yield. Research and methods. Research was conducted in forest area of BKPH Pojok, Kemado Batu Village, Tamakarjo, Grobogan, Jawa Tengah, during March until August 2020. The material using porang plants and liquid organic fertilizer, and the tools using meters, hand sprayer, thermohygrometer, luxmeter, scales, soil analysis laboratory equipment, and stationery. Research design is randomized complete block design or ACBD, 1 factor, 6 level of treatment with each level 6 times. Data analysis using analysis of variance level 5% and regression test. Result and discussion. On the first one is general condition. The type of soil in the research area is little so based on the table data. The results of soil laboratory analysis show that the nutrient content in the soil is low. On the table show that macronutrient content is low, besides that the water content in the land is also low. The affects the growth of bulbil and perennial. And the second is LOF content. LOF content very complex macro and micronutrients as well as microbial content to provide nutrients for the plants. The complete LOF content is able to provide nutrients in littoral soils that have low nutrient content. And the way based on the histogram showed that the treatment that gave the highest average bulbil weight was a dose of 1.125 milliliters per plant. According to Jamila et al. 2002, the role of nutrients in the LF applied influence of the formation of bulbs, number of bulbil, and the based upon the histogram showed that the treatment that gave the highest average bulbil weight was a dose of 1.125 milliliters per plant. And Amir 2021 said that LF application with complete and available component of plants will stimulate photosynthesis, which result in being translocated to all parts of the plant, including to formation of bulbs and the number of bulbs. Tuber first weight. This histogram showed that the treatment at a dose of 1.125 milliliters per plant gave the highest average yield of tuber fresh weight compared to other dose treatment. Willie et al. 2014 said that tuber fresh weight are affected by the content of water and photosynthesis produced from the leaves stored up for tuber formation. Tuber diameter. Based on histogram data, it was shown that the LF dose of 0.375 milliliter plant gave the highest average yield on tuber diameter. Fatirahma and Dodi 2020 said that nutrient calcium affect the enlargement of tuber diameter as a result of cell division and cell expansion and the distribution of photosynthesis. Vardana et al. 2016 said that nutrient kalium is also needed by plants that produce tubers in large quantities because it's needed as a tuber enlargement process. Tuber when dry weight. The histogram showed that a dose of 0.75 milliliters per pound resulted in the highest average tuber when dry weight compared to the other doses treatments. According to FNB 2022, application LF with increasing concentration can stimulate plants to distribute assimilate in the tuber so as to increase crop yields. Arnianti et al. 2010 said that the organic material content in LAF is capable to increase the formation of larger and more tubers. And the last is tuber volume. Based on the histogram shown that administration of a dose of 0.75 milliliters per plant gave the highest average tuber volume compared to the other doses. Application LOF, which has a nutrient content, is able to stimulate the photosynthesis and the result in it being transported throughout the plant by Satyawati et al. 2010.
All right, due to the time constraints and our media presentations guidelines, so I have to stop the ID number 130 for the presentations because it has been more than 7 minutes. So ladies and gentlemen, after we know about the comprehensive presentation from our speakers, it's noticed me that the ginger actually really warm beverage to be drinks, right? Okay, and I like in this chat box, there are no questions here, so we will move to the next sections. And we are welcome to every participant to ask to the speakers via raise hand or chat box. If you want to answer the questions, please note that answer the questions directly using the audio in the Zoom meeting. Okay, so ladies and gentlemen, now we move to, oh, sorry, here there is some questions from Mr. Taufik actually from ONS to ID number 130. The question is, why did you choose organic fertilizers in liquid form instead of solid? So Mrs. Talita, you can answer the questions and turn on your audio. Thank you.
Thank you for the time given to me. I will answer a question from Mr. Taufik. In this research, I chose to use liquid organic fertilizer because it contains more complete nutrient. It is easy to apply and is easily be absorbed by plants because it's in liquid form, while organic fertilizer in solid form are easy to volatile, is easy to leach, and cannot be absorbed by plants directly. The answer is.
All right, thank you, Mrs. Talita, for the nice answers. The first thing is about the easiness of the, what is that, organic fertilizer in liquid form, right? So, okay, due to the times, so we could present the questions from Mrs. Rosmania. So we will directly move to our next sections. Our fourth sections, there will be 3 presenters here with the number ID 155. There will be Mr. Sudadi. Are you here, Mr. Sudadi and team? Mr. Sudadi?
Yes.
All right, Mrs. Triwida Ramadhani. Okay, and then second speakers, there will be with the number ID 179. Are you here, Mr. Nur Ikram Alif Muhammad or team?
Yes.
All right, Mrs. Atika are here. And then the last presenter, there will be with the number ID 205. Are you here, Mr. Ahmad Subaidi and team?
I am here.
All right, Mr. Ahmad and team, thank you very much. Now, ladies and gentlemen, let's welcome together our third presenter. It's time for the operator to play the video.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gents, thank you for the time to me to present our research with the title The Use of Biochar and Biofilm Biofertilizer, Bio2, to Increase Rice Yield. We are from the Soil Science Study Program, Faculty of Agriculture, University of 11 Maret, Surakarta, Central Java, Indonesia. As we know that rice, which is the most strategic food commodity in Indonesia, have to increase the product because of the increase of Indonesian population. And for a long time, the cultivation of rice usually use high dose of chemical fertilizer, such as nitrogen, phosphorus, and kalium fertilizer that can decrease the soil fertility and not support to sustainable agriculture system. And the use of biofilm biofertilizer as environmentally friendly fertilizer will support a sustainable agriculture system for rice. And the use of biofertilizer as a new generation of biofertilizer technology is developed to increase their effectivity, can increase rice yields on soil. And we know that biochar has been widely used with various research in agriculture. So in this research, we will study if biochar can optimize the influence of bio2 on rice yield. Our research aim is to study if biochar can optimize the effect of bio2 to rice yield on algae soil, and also to find out the combination treatment of biochar and bio2 that's keeping the highest rice yield on algae soil. The experimental location is in greenhouse of Faculty of Agriculture, Universitas 11 Maret Surakarta, within the April to October 2021. And the material used is liquid biofilm biofertilizer, coconut biochar, rice variety of IR64, artificial soil sample, and fertilizers like urea, potassium chloride, and sp66 phosphorus. The treatment factor included biofilm biofertilizer concentration with and without biofilm biofertilizer, application method of biochar, spread on soil surface, and mix homogeneously with soil, and the dose of biochar without biochar with 15 ton and 30 ton per hectare of biochar. The experimental design used was a cumulatively randomized design factorial with variable observed included chemical soil properties, plant growth, and plant yield. The data analyzed with ANOVA followed by Duncan multiple ring test if any significant influence. This is the treatment layout, and the result is here. According to the data in the table 1, the fertility of artificial soil use can be classified as low, which were indicated by the low of the soil pH, moderate cation exchange capacity, total nitrogen, total potassium, available potassium, and water dissolved sulfur is low, and the total phosphorus and available phosphorus are very low. The condition of the soil affect the availability of nutrient in it, so that to support soil productivity, it is need to give some input in its management. This is the histogram of the influence of liquid biofilm biofertilizer concentration method of biochar application and biochar dose to rice head at 2 weeks after planting in Alvisual Jumandono. The effect of the treatment on plant head was not significant. The use of Bio2 has not been able to increase plant head, while the use of biochar at the doses of until a certain ton of mixture with the soil accompanied by Bio2 tend to increase plant head linearly, as we can see in the white command. And the effect of the treatment on the number of rice tiller showed similar result, as we can see in 5.2.
All right, the time constraints, I have to stop the ID presenters number with 155 presentations because it has been more than 7 minutes. So sorry, Mr. Sudadi and team. So let's me move to the next presenter.
Assalamualaikum warahmatullahi wabarakatuh, and good day to each and every one of you. My name is Siti Nurul Atika binti Abu Sama from Faculty of Plantation and Agrotechnology, University Technology Merayu ITM, Melaka, Malaysia. Our paper ID is 179 with the title of Effects of Different Organic Fertilizers on Growth and Yield Potential of Solanum melongena eggplant. Solanum melongena is a plant belonging to the Solanaceae family. So it has a rich source of numerous nutrients, energy, sugars, carbohydrate, dietary fiber, and protein. According to the Vegetable and Cash Crop Statistic 2020 by Department of Agriculture Malaysia, the total area planted for eggplant in Malaysia was about 2,385 hectares, and the production was about 39,785 metric tons per year. So Solanum melongena is a vegetable crop where its growth rate and yield quality are largely influenced by fertilizer application. Currently, chemical fertilizer is widely used in agriculture, including vegetable crop planting, because of its high productivity in a short period of time, despite of its drawbacks such as expensive and resulted in nutrient imbalance and soil acidity. So the application of organic fertilizer to crop has high economic advantages when compared to inorganic fertilizer. This organic fertilizer application can significantly enhance growth and yield quality of Solanum melongena fruit. So the different organic fertilizer could help to alleviate the significant reliance on chemical fertilizers while lowering the cost and improving eggplant growth and output. Therefore, the objective of this study were to determine the growth of eggplant and to quantify the fruit yield based on the fresh weight as affected by different organic fertilization. So for the materials and method, the experiment was conducted in greenhouse 3 variety Melaka branch Jasin campus. The selected variety was Antabago hybrid F1. So the experimental design used was LCBD with 4 replication. Planting container used was polybag, and the media used was topsoil and sand. For the treatments, there were 4 treatments with four replication. The first one was T0, no fertilizer, which acted as the control. T1 consists of chicken manure, T2 consists of vermicompost, and T3 consists of biochar. So there were 2 application of treatments. So for the first time was applied during the soil preparation, and the second application was given after 4 weeks of the transplanting process. For the data collection, there were 2 types of data were being collected. The first one was growth data, consisted of plant height, number of leaf per plant, leaf area, and total of plant fresh weight and dry weight. The second data was yield potential data, which consisted of number of flowers, number of fruits, fruit fresh weight, and fruit dry weight. As for for the results, we go to the growth data. So based from table 1 and table 2 showed here, the plant height, number of leaf area, total of fresh weight and dry weight of plant showed significant differences compared to the other treatments on chicken dung. So this result was similar to the previous study by other researchers, where application of chicken dung resulted in the highest plant height, number of leaf area, fresh and dry weight of plant. So this chicken dung fertilizer that enters the soil was decomposed by microorganisms, and plants will absorb the nutrients that occur from the decomposition process and thus increase the height of the plant. So the similar thing happened to the number of leaf, where this chicken dung will promote the assimilation of the nutrients to the plant by increasing the absorption and transport of nutrients by the roots, which can increase nutrient absorption for leaf formation. As for the leaf area, so when chicken dung was used, it supplied nutrients to the growth of leaf size, which increased the leaf area and in turn increased photosynthesis to give energy to the plant to support the plant growth, so which eventually increased the total fresh weight and total dry weight of Solanum melongena. For the second data, which was for the yield data, as you can see from this table, for the number of flowers, chicken dung showed highest significant differences compared to the other treatments. However, for the number of fruits, fruit fresh weight, and fruit dry weight, there's no significant differences between treatments. So for the number of flowers, this result similar to the other studies by other researchers, since the nutrients in chicken dung promote high vegetative growth and ultimately increase during the flowering stage. So that's why the number of flowers is increased when chicken dung was applied to the treatment. But however, for the other parameters, although it showed no significant differences, but the highest mean data was shown on the chicken dung application compared to the other treatments. So for the conclusion, the present study confirmed that chicken dung was the best organic fertilizer for eggplants growth and yield production. So therefore, chicken dung is recommended in the cultivation of eggplant as an alternative to the other organic fertilizers or chemical fertilizers. The usage of chicken dung, which can reduce environmental contamination, especially water pollution from agriculture from chemical fertilizer usage, and its lower price would definitely help in reducing the cultivation costs for eggplant. So with that, I end my presentation. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. We would like to present our work entitled Optimism, Not Lombok. Sorghum is, although it has a easy and a lot of uses, it is less attractive to farmers due to the low selling of availability of local markets. One of the efforts to introduce more sorghum is by intercropping with the main crop in an area without reducing yield of the main crop. Peanut is a secondary crop that is widely planted in North Lombok in crop rotation with the low land rice system. In addition to the high selling value of the product, peanuts actually have a very good agronomic and conservation impact on the soil and plant. an effort to introduce sorghum cultivation, intercropping with peanuts and soybean has been attempted. Agronomically, the intercropping is profitable, but resulting lower yield on the main crop. Therefore, it is necessary to arrange plan in terms of population size or planting time of sorghum. Our experiment was carried out in the field on the dry land in Ganga district, North Lombok. The experiment was conducted from May to August 2021. The treatment of plant arrangement is inserting one row of sorghum, after one row of peanut, after 2 rows of peanuts, 3 rows of peanut, 4 rows of peanut. While sowing time consists of sowing peanut 14 days before sorghum, sowing at the same time with sorghum, and sowing 14 days after sowing sorghum. From the experiment, we can see that the highest yield we get for peanut is if we sowing peanut before sorghum, which is 1.21 ton per hectare. While on the plant arrangement, we get the highest yield is if we insert 1 rows of sorghum after 4 rows of peanuts. It is 1.18 ton per hectare. I said that sowing peanut before sorghum give the best yield and yield component compared to peanut sowed at the same time with sorghum or after sorghum. While in the treatment with sorghum insertion, the highest yield of peanut was obtained at the loosest insertion, which is 4 rows of peanut inserted with 1 row of sorghum, which could be due to the fewest competitor and the highest population of peanuts. If we look at the data of sorghum, we found that the highest yield is if we sowing at the same time and before peanuts, and if we sow sorghum at the same population with peanut, which is 6.8 ton per hectare. So sorghum sowed before peanut give the best yield. There was decrease in yield of sorghum if we sowed after peanut. Peanut yield was so low at the sowing after sorghum, showed a decrease in the yield on the 2 plants component in intercropping. Sowing sorghum between 4 rows of peanuts give a yield around 4.81 ton per hectare, or is about 60% of the potential yield. Peanut sowed in 4 rows with 1 row of sorghum yielded around 1.21 ton per hectare, which is 80% of potential yield. So our conclusion that peanuts sowed before sowing sorghum give the higher yield compared to crops sowed after together or after sowing sorghum. Plant spacing or population has an effect on crop yield. The highest yield of peanut is obtained by inserting 1 row of sorghum between 4 rows of peanut. The best result in this intercropping peanut sorghum is by sowing peanut 14 days with sorghum and inserting 1 row of sorghum between 4 rows of peanuts. That's become our suggestion that if you want to introduce sorghum to farmers, planting in intercropping with peanuts can be done by sowing 1 row of sorghum between 4 rows of peanut. Sorghum is sowed 14 days after sowing peanuts. Thank you very much. That's what I can say.
Okay, thank you very much for all presenter. It is very interesting presentation, of course. We know that Solanium melonga is a high of antioxidants, so that's why it must be planned properly to have high in production. Chicken dung can be the best organic fertilizer, of course. And now, wow, it is very interesting. In the chat box, there is Mrs. Talita and Mrs. Rosmania discuss about the POC, and it's really interesting, we think. But so sorry that for the time limitation, we can accommodate the discussion at the previous sections. And now in these sections, in the chat box also, there is questions from Mr. Zulfahmi, to ID number 179. The question is, why is the value of fresh weight eggplant using chicken dung very high compared to the other treatments? Is the value really? Okay.
Thank you, Mr. Zulfahmi, for the question. Okay, for your information, the value is and when we use the chicken dung for the organic fertilizer, especially when we try to find the data by using the total plant fresh weight and total plant dry weight, we have to include all the plant parts into the total of the plant fresh weight. So when we include all the plant parts, including the fruits, flowers, and the parts, so it showed that it has the highest number of fresh weight compared to the other treatments. So that's why the value is comparatively higher compared to the other treatments.
All right, Mr. Zulfahmi. So sorry, all of audience, I have troubles from signals just now. So for Mr. Zulfahmi, is there any questions or feedbacks for Mr. Atik or Mrs. Atika? All right, so sorry. No one? Okay. Okay, is it okay for my signals, everyone? Can you hear my voice over there?
Yes, okay.
Yes.
Okay, hello everyone. Can you hear my voice? So sorry, I have trouble with my signals the time before.
I can your
Okay.
voice very well.
All right. Okay. Thank you, Mr. Sudadi. Thank you, Mr. Zulfahmi. No feedback, not clear. The answer, trouble signals. All right. Yeah. Yeah. So sorry for that. Okay. Thank you very much for all presenters in every section and team for giving such an informative and comprehensive presentation. Of course, ladies and gentlemen, in the chat box, our commit had been shared to all of you guys, the attendance form. Please kindly to fill the attendance forms now. We are going to have a short refreshment for a minute. You may have a cup of tea and stretchings maybe, or you have some food to eat. And please return to this room at 10 to 20 times Jakarta's time, and we will continue with the fifth and section presentation until finish. Presenter certificate information and another informations will be shared in the end of this session. So please, having a refreshment times, you can have a cup of tea or a cup of coffee, and we will back in 10 to 20. Okay. Baik, Bapak Ibu, saya sampaikan dalam bahasa Indonesia. Sebentar lagi kita akan menghadirkan waktu untuk istirahat bagi Bapak Ibu semuanya. Kita istirahat dari sampai jam 10.20. Jadi nanti jam 10.20 Bapak Ibu harap kembali ke ruangan ini lagi dan kita akan melanjutkan presentasi. Ya, terima kasih.
Maaf Bu Nurul.
Ya, siap. Pak Sudadi,
saya mohon
bagaimana?
izin ya setelah ini ya, karena saya ada di apa namanya CBI Pangkalan Bun ini. Saya ada tugas ini setelah ini ya.
Siap. Bisa digantikan dengan tim yang lain Bapak untuk bisa
ada anggota kami Triwida dan Profita. Terima kasih. Assalamualaikum.
Siap. Matur nuwun Bapak. All right, ladies and gentlemen, we will start the next sections in about a minute. So I will have to make sure that all of the operator here is ready to start this section. Mr. Gale, are you ready for that? Hello, Mr. Gali. I think Mr. Gali still having a cup of coffee. So we have to wait for a minute, ladies and gentlemen. Okay, we think that Mr. Gali has been ready for that. So ladies and gentlemen, welcome back to our next section, the parallel session of the 9th International Conference on Sustainable Agriculture and Environment, Universitas 11 Maret Surakarta, 2022. The rule is same as previous section, of course. In every section, we will play 3 videos from our presenters, then followed by discussion with the presenters. Now, we are going to watch together the video presentations from our best presenters with the number ID 7. Mrs. Elvi Rahmadani, are you here in this room, Mrs. Elvi? Mrs. Elvi or Mrs. Hoyeria, are you here in this room? All right, I think Mrs. Elvi is here. Okay, thank you, Mrs. Elvi. Yeah.
Yes, thank you very much.
Okay, and then second speakers will come to with the ID number 25. There is Mr. Ronnie Junior. Hello, Mr. Ronnie Junior. Are you here in this room? Mr. Ronnie Junior and team? Okay. Okay, we have to skip this presenter because the presenter is nothing. So we will move to the last video in the 5 sections. There will be with ID number 52, Mrs. Siti Rohmah.
Yes, I'm here.
Okay, thank you, Mrs. Siti. Now, let's we watch together. Welcome to the operator to play the video.
Assalamualaikum warahmatullahi wabarakatuh. Good morning. I'll to present my paper, the title The Effect of Collaboration Between Elements for the Progress of Rubber Cooperative in Riau Province, Indonesia. My team are Koiria and Anistatik Mariani. Riau is one of potential province for smallholder rubber rejuvenation activities funded by Indonesian government. However, this program is not contribute evenly to all members of the rubber cooperative. That we know, the rubber cooperative is an important agency to support the economy of the rubber farmer group in Riau province, but the number of active cooperatives declined since 2014 until 2020. You can see the report from the table my presentation. It's a great important to analyze further about the collaboration of cooperative element against. So the goal of this study is to assess the aspect of collaboration between elements against cooperative progress. This research was examined both inactive and active cooperative as a comparison. Material and method. Research sites. The study was assessed on active and inactive rubber cooperative in Kuantan, Singingi, and Kampar Regency as the rubber center location in Riau Province. The selection of cooperative was conducted by talking three groups of inactive rubber farmer cooperative and three active one as a comparison based on the data from the Kuantan, Singingi, and Kampar District Trade Cooperative Office and submitted in 2016. Data collection. These research data are primary and secondary data. Primary data gained by direct interview with informant and respondent as the research sample, namely management, supervisor, employee, and cooperative member. And secondary data was collected through written data in the field or related institution as applied in Rahmadani 2020 and 2021. Population and sample that research means all stakeholders in rubber cooperative selecting both of active and inactive cooperative in Kuantan, Singingi, and Kampar Regency. There are three selected active cooperative are KUD Harapan Rakyat, Koperasi Maju Basamo, Koptan Karet Sumber Rezeki, and three inactive cooperative are KUD Lestari, KUD Harapan Rakyat, KUD Bukit Songgang, as shown in table in previous report Rahmadani 2020-2021. This is a table population and sample. 50 sample we taken river to Kohen, Bali, and Rescue. Data analysis. The interview data in this study we collected from the perception of each internal element in the cooperative ensuring with scale of 1 until 5. Data were also canvassed by Excel from the indicator of the collaboration among the cooperative internal elements, member, management, supervisor, and employee. As shown in figure 2, the assessment of the parameter represented by X reference indicator from Robin 2001, the value was grouped into 5 categories as applied in Rahmadani 2020 and 2021. Okay, we can see the result on discussion. The result present 6 main topic of discussion. The first result we can see the table 1. The table explain for we collaboration between member management as a good score of active cooperative. The score 77.60%. Then indicator we can see at figure 2 60.76% until 89.61%. The collaboration that I mean is a form of social process that occur between cooperative elements in the interaction to responsibility in data contribution and solidarity. Different with in active cooperative, we looks in inactive cooperative has a fairly good category value with score 44.61%, and all indicator we can see the figure 2 59.12% until 47.23. You can see the figure 2, the collaboration between member and management of rubber cooperative in Riau province. We can see figure 2 can explain the source of problem come from the key 3 indicator with the lowest value. Another problem come from the first, the obligation aren't carried out optimally by cooperative. For example, the RAT report is not complete optimally and many members are in error, don't pay mandatory saving. Next, collaboration between member and supervisor. The collaboration between member and supervisor has a good interaction, score 78.42%, with the indicator value in a good, very good category, 74 until x1%. Of the X indicator of cooperation aspect, it shows that there are problems between member and supervisor. The short problem is in key 3 and key, while in the active cooperative there are still problems from the interaction because it has a fairly good score 40 4.6. Namely, the application of rules and sanction together score. Okay, we can see the collaboration between management and internal element of our farmer cooperative, supervisor and employee. That we can see the table show the collaboration between management with supervisor Act 4 and management employee Act 5. It has a good and very good value for active rubber farmer cooperative. The score 79.30% and 80.06%. A bad and good enough value score 40.00% until 47.2% were found in an active cooperative. This program in an active cooperative interaction between Act 4 and Act 5. Figure 5 looks the ability to cooperate between management and supervisor in an active rubber farmer cooperative. Has a very good interaction values. The score 80.06% with indicator from the
All right, due to the time constraint, I have to stop the ID number 7's the presentations because it has been more than 7 minutes.
Okay, sorry.
Okay.
Assalamualaikum warahmatullahi wabarakatuh. Today I would like to present my research entitled Factors Influencing Biological Asset Disclosure in Agricultural Companies in Indonesia. My name is Siti Rohmah Ika. This is our research together with my colleague Romi Susetyo, Angelia Pribadi, Titok Dwi Narno, and Ari Kunchara Widakdo. We come from Janabadra University and 11 Maret University. This is the introduction. Agricultural companies are those whose activities relate to managing the biological asset transformation of living plants or animals. It calls biological asset into a product that ready for consumption or which still require further processing. So the biological assets, for example, is animal or plants. According to the Indonesian Statement of Financial Accounting Standard 69 or PSAK 69, agricultural companies should report in how they recognize and measure the fair value of their biological assets in the company annual report. The reporting is effective from 1 January 2017 and it revised to 1 January 2018, but early implementation is allowed. Our research objective is to examine to what extent the biological asset reporting according to the PSAK 69 among agricultural companies listed on Indonesia Stock Exchange or IDX. This research also examined factors that may influence the level of biological asset disclosure in the company annual report. So the examined factors are biological asset intensity, concentrated ownership, audit committee effectiveness, profitability, and company size. This is the research method. Our sample is agricultural companies that listed on IDX in 2017 to 2019 consecutively. The total numbers is 36 companies. This is the variable measurement of biological asset disclosure. This consists of a checklist that derived from PSAK 69. This is an adoption of IAS or International Accounting Standard 41 about agriculture. So the index is measured in yes or no items with a total of 40 items of disclosure and it expressed in percentage. This is the measurement of independent variable. The biological asset intensity is the sum of the biological assets divided by the total asset, while the managerial ownership is a number of shares owned by the management divided by the total outstanding shares. Audit Committee Effectiveness Index is an index of audit committee effectiveness based on ICA and Gazeli 2012 index, and the control variable is profitability and size. This is the descriptive statistic of the all variables. As you can see, the mean of biological asset reporting is about 80%. It is relatively high. The mean of biological asset to total asset is 38%. The mean of ownership concentration is 75%. So the agricultural companies in Indonesia stock exchange is highly concentrated. Meanwhile, the profitability is 3.4% on average. This is the regression result. As you can see that the significant variables is biological asset intensity positively influence the biological asset disclosure, significant at 1% level, and size also positively influence the biological asset reporting, significant at 5%. So this is the conclusion of the study. The average level of biological asset reporting is about 80%. Biological asset intensity and size are positively associated with biological asset reporting. It means that the higher the number biological asset in a company, the higher also biological asset reporting in the company annual report. The bigger the size of a company, the higher also biological asset that are available in the company annual report. This is the limitation and future research suggestion. The period of observation is 2017 to 2019, that covering the effective date around the implementation of such report according to PSAK. However, this research does not examine the different level of disclosure before and after the effective date of the disclosure. So future research might test the difference either pre and post the effective date of implementation or pre and post COVID-19 impact on the disclosure. This is the last of our presentation. Thank you, terima kasih, and arigatou gozaimasu.
All right. Thank you very much for the great presentation for all of the presenters here. Ladies and gentlemen, just because the ID number 25 can attend in this room, so we will directly move to the discussion. Okay, first I wanna make sure that the ID number 25 is not here. Hello, Mr. Kantingan. Sorry, Mr. Ronnie. Okay, yeah. Okay, so we will directly move to the discussions. And here actually there are no questions in the chat box, but I want to ask to the ID number 7, Mrs. Elvi Ramadhani and team. Okay, because she cannot finish the presentations before, so I want to ask, what is actually the conclusions of your research, Mrs. Elvi?
Hello.
Yeah, I can hear your voice. So actually, what is the conclusion of your?
The conclusion that my research collaboration arising from the element interaction greatly affect the progress of rubber cooperative in Riau province. Active rubber cooperative are able to collaborate well as proved by good until very good. Value score 72.43% until 83.51%, and inactive cooperative bad value enough, bad value until good enough, value the value 20% until 47.2%. So in this aspect, inactive cooperative inform that collaboration problem inhibits the cooperative progress. The main problem come from the interaction between members and employee that we call the symbol X3, and management with employee X5, and supervisor with employee. We say that we give the symbol X5. Okay, X6. Yeah, sorry, I'm forget it. Okay, thank you, moderator. This is my conclusion.
All right, thank you very much for the explanation, Mrs. Alfie.
Yes.
Okay, so
I'm
ladies
sorry,
and
my
gentlemen,
presentation is to be long.
it's okay, it's okay,
Yeah.
it's okay, Mrs. Alfie. All right, ladies and gentlemen, I will remember again that we are welcome to every participant to ask to the speakers. If you want to answer the questions, please answer in the discussion time directly using the audio. And if you want to ask to the speakers, please raise your hand or chat in the chat box in the Zoom, of course. Okay, ladies and gentlemen, now we move to the second sections. Allow me to welcome for our three presenters. There is number ID 61.
Yes,
You
I'm
will
here.
say hello.
Yes,
All
hi,
right,
I'm here.
Okay, right, Mr. Ahmad. And then for the second presenter, there will be with the number ID 73, Mrs. Rosmania. Are you here, Mrs.
Rosmaina, I'm
Rosmania?
here.
Rosmaina, okay,
Yes.
sorry. Okay, you look so beautiful today, Mrs. Rosmaina. All right, and then the third speakers will be with the number ID 89, Mr. Muhammad Faris Saiman. Are you here, Mr. Muhammad Faris?
Yes, I'm here.
All right, so please, all right, thank you, Mr. Faris. Please, we will come together for the operator to play the video presentation.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and a very good day to all the panels of the International Conference on Sustainable Agricultural and Environment. My name is Ahmad Kumal Benusli from Faculty of Plantation and Agrotechnology, University Technology Marang, Malaysia. Today, I would like to present on the title of a review participation of cocoa smallholders in an agricultural certification scheme. Basically, we know cocoa is the most famous crop that are globally known. But in order to ensure the productivity of the cocoa which can fulfill the market demand, the answer is agricultural certification scheme, which can help to boost the productivity of the cocoa crops. Let's check it out. Without further ado, now let's take a look on the first phase of this presentation. Cocoa, or known as the Tribuma cacao else, is the primary commodity crop in Malaysia, like oil palm, paddy, and rubber. Cocoa comes from the Malvaceae family. In the tropical world, cocoa is a very significant crop that are widely used in the manufacturing of chocolates and other end products. In Malaysia, Malaysia Cocoa Board will manage the cocoa industry under the Ministry of Plantation, Industry and Commodity in the way to make the Cocoa Malaysia become more revival and essential crop throughout the year. If you want to know, cocoa crop has been steadily expanding over the last few decades in Malaysia and also in the world. Because of what? Because of the organized policy support, crop area expansion, and also the good yield of the productivity, the crop area. Based on the Food and Organization of United Nations, FOSTEC stated that Malaysian cocoa crop at the ranking number 13 in the world, with the production of 1,005 ton in the 2019 that contribute to the 1.64 billion ringgit Malaysia of the gross domestic profit GDP on the 2020. Generally in Malaysia, there are three places that produce the crop area. For the first one is the Peninsular Malaysia, that include Kuala Selangor, Bagan Datuk, Sabak Bernam, Muar, Temerloh, and Batu Pahat. For the second area is the Sarawak, and for the third area is Sabah. Based on the data from the MPIC Malaysia, cocoa crop in Malaysia at the ranking number 3 in the agri-commodity sector that produce the cocoa crop around 227 tons, which include the 3 areas in the Malaysia that produce the cocoa crops. So, this study is to review and measure factors and benefits of the smallholders' participation in the agricultural certification scheme. Generally, agricultural certification scheme, there are several certification either locally in Malaysia and internationally that must match the cocoa sector requirement in order to support the producer groups and cocoa smallholders. If you want to know, based on the MPIC Minister Yang Berhormat Datuk Zuraida Kamaruddin claimed that the country cocoa sector is a well-known and the production of the cocoa is keep growing throughout the year. Now let's take a look on the method that I used in the review study. For the study selection, the article published were searched from the Web of Science, Scopus, and also Google Scholar. For the searching part, I using several keywords such as agricultural, certification, scheme, factor, influence, motivation, adoption, and many more. The last part of the method is the data extraction and analysis. The article that meets the review criteria were found by using the comprehensive search to define the factor and benefits of this study. Now, let's take a look on the major part of this study. They have several studies identify the key elements that influencing the smallholders' participation in the agricultural certification scheme. Now, let's take a look on the first part is supporting the smallholders is the factor that affecting the smallholders' participation in the agricultural certification scheme. A study was found Malaysia Cocoa Board will provide cocoa smallholders with a good motivation and support, including the planting supply, raw material, and provide a lot of information and knowledge to the smallholders. Other than that also, a productive MCB staff under the Department of the Extension Agent will ensure and provide the information regarding the new technologies to the smallholders. Next is the certification awareness. Certification awareness is typically a factor that affects cocoa certification among smallholders. Participating smallholders in certification program are overwhelmingly positive about the program advantage. Moreover, the smallholders' involvement in the training and courses will impact whether or not they receive certification on a national or worldwide level. The willingness of the certificate is encouraged by the knowledge of the significance of certification and the procedure required for any crop, particularly in the cocoa sector. Therefore, if a farmer have more knowledge and awareness, they will be more eager to obtain the certificate. Additionally, the findings stated that attitude could be one of the influencing aspects to the smallholders for the certification based on theory of plant behavior. Furthermore, age in agricultural certification scheme choice is consistent with a study that has been conducted in Kenya, which found that the age of smallholders is favorably related to the decision to participate in a certification scheme, especially in the agriculture sector. Now, let us take a look on a last part of the factor that influencing the agricultural certification scheme among the smallholders, which is in determining an individual quantification and identification of their preferences over a collection of options. A study was claimed that the theory of preferences is a crucial essential component of individual decision behavior. Now, let's move to the last part, which is benefit participating in agricultural certification scheme. Participating in agricultural certification scheme will be as a guideline that has been successfully strategy for easing the transition to goods and sustainable agricultural production. A study found that the price for certified crops are higher than uncertified crops. They get a lot of training, especially in terms of good and agricultural practices. Other than that, the extrinsic and intrinsic reward that employees receive can be affected by the certification scheme, which might affect a job satisfaction. The certification scheme prioritizes smallholders' empowerment and the bargaining process to strengthen the guidelines for collective agreement between the labor and the traders. Hence, the certification scheme delivers many advantages to the smallholders in developing countries with a good system. The certification aims to increase the consumer awareness
All right. Actually, it is a great presentation that comes from University of Technology Mara, Selangor, Malaysia. But due to time constraints, I have to stop the ID numbers 61 for the presentation because it has been more than 7 minutes. So sorry for
Sure.
that. All right, ladies and gentlemen, let's move to ID number 73.
Good afternoon, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. I am Rosmaina from the Department of Agrotechnology, Faculty of Agriculture and Animal Science, State Islamic University of Sultan Syarif Kasim Riau. Today, I want to present our research report with the title Pineapple Genetic Diversity in Riau Peatland Assessed by Random Amplified Polymorphic DNA Marker. This is our team: Nugroho Febriandi, Ervina Arianti, Rita Elvianis, Nila Hayati, and Mr. Julfahmi. Our background of research: pineapple played an important role in the global fruit trade. Indonesia is the fourth largest pineapple production country in the world after Philippines, Costa Rica, and Brazil. Riau is one of the centers of production of pineapple in Indonesia. The of pineapple cultivated in Riau are queen pineapple and smooth cayenne pineapple. Pineapple cultivation in Riau is carried in monoculture and intercropping with coconut, palm oil, and rhubarb. Pineapple are heterozygous plants that have a cross-pollination system. This happens because pineapple have soil-incompatible properties, so that crosses can only occur between different cultivars. Previously report that there was morphology diversity on pineapple in Riau province. This study aims to determine the relationship and genetic diversity between local variety cultivated in Riau peatland based on random amplified polymorphic DNA marker. In this study, we used 10 pineapple genotypes from 5 districts in Riau, namely Paun, Queen from Siak, Gemilang, Nanas Bangka, Bubur, Queen, Mahkota, Queen from Bengkalis, Suska Kualu, and Queen from Teluk Makmur. Stage of labor, DNA extraction, electrophoresis, and then PCR, electrophoresis for DNA amplification, UV illuminator, and data analysis. At the beginning of our study, we used 18 primers for primer selection. There were 11 primers capable of amplifying DNA bending pattern. Result and discussion. The result of the amplification of 11 RIBD primer obtained 76 loci, of which 96.42% were polymorphic loci, with a band size of 300 to 1500 base pair. Furthermore, the amplified date was used to see the relationship between 10 pineapple genotype T. This is DNA amplification, and this is a dendrogram based on 73 polymorphic band on 10 adhesion of pineapple. Based on the UPGM dendrogram, the coefficient similarity between the test genotype rank from 0.53 to 0.91, or has a level of diversity of 0.47 to 0.09. At the coefficient similarity of 0.70, all pineapple are grouped into 3 main groups. The first group consisted of 2 genotypes, namely Faun and Bangka from Tembilahan, both of which were smooth cayenne cultivar. The third group consisted of 7 genotypes belonging to the Queen cultivar. The second group only consisted of 1 genotype, namely Gemilang variety. It can be seen that Gemilang is between the smooth cayenne and queen group. Alleged natural crosses between cultivars are very possible because at the location of this study there were 5 pineapple genotypes, namely Bangka, Paun, Queen, and Bubur, which were planted simultaneously in one garden. The result of this study indicated that RVD can identify and separate the queen and smooth cayenne in different groups clearly, and the large variety gemilang is the result of natural crosses of queen and smooth cayenne clearly separated. This is the morphology of queen and smooth cayenne, and then gemilang. The leaf and color of fruit, flower, and conclusion. Based on the result of this study, it can be concluded that the relationship between pineapple genotype in Rio ranges from 53 to 91%, which means that there is a diversity of 47 to 0.09%. Based on the UPGMA dendrogram, it can be seen that queen and smooth cayenne cultivar are separated groups and confirm that gemilang variety is the result of natural cross of smooth cayenne and queen pineapple. Thank you for your attention.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Faris bin Saiman from University Technology Mara, Jasin, Malaysia. Today, I will present my research which is chelated copper zinc as potential for foliar fertilizer based on different pH and electrical conductivity. Without wasting any more time, let us continue to introduction. Micronutrients such as copper and zinc are very important in the growth of plants. Micronutrient deficiency are widespread in many Asian countries due to calcareous soil properties, low pH, salt stress, constant drought, and unbalanced use of nitrogen, phosphorus, potassium fertilizer, RIAS 2020. Some of the adverse effects of stress caused by micronutrient deficiency on crop include low crop yield and quality, imperfect plant morphological structure such as small xylem vessel, widespread infection of various diseases and pests, and low activation phytocedospores and lower efficiency of fertilizer use. When the plant combined with adequate plant nutrient, it can improve quality and quantitative of plant products. Due to their involvement in enzyme activity, copper and zinc in the nutrient supply of soil and plant should be prioritized for the basic microelements. Their deficiency greatly affects the quantity and quality of crop product. Narimi 2010. Next, this research was conducted by using pineapple plants. So there is a little bit information about pineapple. Pineapple cultivars grown in Malaysia consist of 3 large groups, which are cayenne, Spanish, queen, and the other one we have hybrid pineapple. Pineapple is a plant from crassulacean acid metabolic, CAM group, same like cactus plant. So the advantages of these plants are capable of making photosynthesis with low water availability. The stomata are closed during the day, meaning that when we want to apply foliar fertilizer, we must apply it in early morning or late evening. Lastly, this plant can adapt in low pH soil, for example in peat soil. According to Beltagi, copper and zinc are necessary micronutrients for plant physiological and biochemical process. Copper deficiency can cause light green, narrow leaf with wavy borders, have U-shape in cross-section, and a stunted plant, while zinc deficiency can cause chlorosis at beginning of exposure in the younger leaf, and the chlorosis will extend it to older leaf. Next slide is methodology. Determination of pH and electrical conductivity of different rates of copper and zinc fertilizer. This research was conducted at University Technology Maharajasin Laboratory. 6,407 until 2,400 ppm of copper and zinc EDTA will be prepared in a different beaker, meaning that there will be 6 beakers with different concentration of copper and zinc. Next, 8.89 gram of nitrogen, 3.86 gram of phosphorus, and 2.18 gram of potassium will be diluted with 250 milliliter of water. Then, NPK solution will mix with copper and zinc solution. The pH and EC of the solution will be observed by using pH and EC meter. Next experiment is pineapple cultivation. This study was conducted at coordinate 1.842162 103.142060, which is in Malaysia. I will use Milidilate MD2 pineapple, which is hybrid pineapple. The sucker will be dried under direct sunlight for 3 days to remove the pest and excessive water in the plant. Sucker will be cut to 35 cm height and will be planted in polybag with double row spacing system with 45 cm x 60 cm x 90 cm, 1 feet x 2 feet x 3 feet. The plant will be arranged according to randomized complete block design RCBD with 5 replications of a treatment. Next, this is the treatment. There are 5 treatments, which are treatment 1 control, treatment 2 NPK fertilizer, treatment 3 6,407 ppm of copper and zinc plus NPK, treatment 4 7,208 ppm of copper and zinc plus NPK, and treatment 5 is 8,009 ppm of copper and zinc plus NPK. I use 6,407 ppm until 8,009 ppm as the 3 best treatment because the suitable pH and EC is 5 until 7 and 1.6 until 2.4 respectively for plant uptake. Next slide is result. The fertilizer rate had the effect over the pH and electrical conductivity. The fertilizer rate in 8,009 ppm, which is treatment 5, show highest rate for plant growth, but it require more fertilizer compared to treatment 3 and treatment 4. The fertilizer rate in 6,407 ppm, which is treatment 3, show lower rate for plant growth among treatment 4 and treatment 5. However, it's still better from control plant. The ANOVA test revealed that the fertilizer rate had significant effect on pH and electrical conductivity by p-value below than 0.01. Next slide is discussion. It was observed that the plant growth with various concentration of copper and zinc level different in plant growth. This study shows that the use of 8,009 ppm of copper and zinc chelated fertilizer is the best choice. It will produce a pH of 6.74 and EC of 2.403. This is in line with the optimum value for pH, which is 5 until 6, while for electrical conductivity is 1.8 until 2.4. Studies shows the higher rates of fertilizer, the pH will be more alkaline and the electrical conductivity will be the higher. Second, plant growth was increased with the increase in the pH and EC or with the increase of copper and zinc concentration, means that the optimum nutrient uptake would have caused such improvement. Conclusion. This paper focused mainly on provide insight on the use of copper and zinc chelated fertilizer, especially to assist farmers in increasing their agriculture yield. This paper discussed how the use of copper zinc chelated fertilizer can influence the growth of plant, which in turn will produce healthy and fertile tree. This research aimed to investigate the influence of copper zinc mixture in different rates on pH and electrical conductivity as a potential for foliar spray fertilization. This requires various strategies such as the use of chelated fertilizer to strengthen the value chain, especially on agronomic practice using mineral soil to ensure the sustainability of production. Based on the finding above, it can be concluded copper zinc chelated fertilizer mixture at different rates will affect pH and electrical conductivity. The rising cost of fertilizer will burden farmers in all over the countries Therefore, the use of copper zinc chelated fertilizer should be used to increase the plant growth and the yield of farmers, and in turn can improve the living standard of farmers. That's all from me. Thank you very much.
All right, it's a good presentation for all of our presenters here. I also believe that Malaysia really rich in the cocoa production, the most cocoa production from Malaysia that I know most is about the Naraya oat cocoa and Rocky cocoa. Is that right, Mr. Mr. Ahmad Lukman?
Yes, correct, doctor.
Yeah.
Cocoa is a very
Yeah.
interesting product, right? Which is mostly all the people will like to eat cocoa. So, doctor, they have one question in the chat box that.
Yep. Okay, right. This is there is one question from Mrs. Elinor to the ID number 61.
Yes,
What
for
are
me.
the benefits for cocoa farmer in the agriculture certification scheme? Okay, please answer, Mr. Ahmad Lukman.
Okay, thank you to the Madam Elinor for the question. What is the benefit for cocoa? What is the benefit for cocoa farmers in the agricultural certification scheme? So basically, they have a lot of benefit because when the farmers, when the smallholders or farmers apply the certification scheme, for example, the farm gate price. The farm gate price will be maintained. For example, when I give you one example, when the price for the local price is, for example, 5 ringgit, when they apply the agricultural certification scheme, the price will increase to 10 ringgit. But when the market price at the international market trader will reduce, the price will be maintained at 10 ringgit. This will maintain the price of the cocoa certificate when they apply the cocoa certification scheme. Other than that, also they also will give the environmental and social condition impact, which is the Malaysian Cocoa Board under the Ministry of Plantation Industry and Commodity will delivering a good system to the smallholders. This is one of the benefit, can increase the consumer awareness in the way to ensure the welfare effect of the cocoa smallholders also will be taken care by the Malaysian Cocoa Board. Other than that, also it also will increase and premium up the supply chain in order to demonstrate the anchor cooperation effective and also to gain a new market share. When the smallholder apply the agricultural certification scheme, especially it will increase the market share, and in order to ensure that the product that has been produced by the Malaysian Cocoa Board and also the Ministry of Plantation Industry Commodity can fulfill the market demand, and also we can sell our product into the international market traders.
Okay.
All right. What a nice and comprehensive explanation from Mr. Ahmad. So, Mrs. Elinor, there is feedback for Mr. Ahmad, or it is enough?
Okay.
All right. So, thank you very much, Mr. Ahmad. Oh, sorry. Okay, so ladies and gentlemen, now we will move to the next sections. There will be 3 speakers again in these sections. I will call for the ID number 91. Mrs. Aida, are you here in this room, Mrs. Aida? Mrs. Aida, Halida, and Tim, are you here in this room? Sorry, Mr. Elinor, can you mute your microphone first? Thank you.
Okay.
Yeah. So we will move to the next section. Mrs. Aida Halida and Tim, are you here in this room? I think. Okay. Okay, we will move to the next presenters. ID number 104. Mr. Panji Harjanto. Are you here in this room, Mr. Panji?
Panji.
Mr. Panji, Mr. Imam. Sorry, Mrs. Elinor, can you mute your microphone, please?
Ya, oke.
Okay, finish. I have mute your microphone. Okay, Mr. Panji and Tim, are you here in this room? All right. For the third presenters with the ID number 138. Mrs. Jati Widunu, are you here in this room?
It's
All right.
Mr. actually.
All right, so sorry, Mr. All right, Mr. Jati Widnu, I will give time to make presentations or play the video for the operators. Welcome to the operators to play the video presentations.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. My name is Aida Halida binti Hamdan. So today I would like to present my research on the morphological characteristic and gamma irradiation effects on plant growth of stevia ribaudiana. Okay. Now we proceed to the introduction. Okay, there are so many countries that are now commercially cultivating this stevia plant. All major regulatory agencies around the world have approved or adopted high purity stevia glycoside as sweetener, and more than 150 countries has approved or adopted their use in food and beverage. Stevia has the sweetest flavor and contain other biochemicals that are beneficial to humans. It is bitter perennial herb with a large root system that produces thin elliptical leaf. Nowadays, cultivated plants are said to be more common. The leaves are the most useful component of this plant. But the 18 species out of 110 tested for sweetness were found to have this trait. The seeds are extremely small. Since the seeds are so small, seedlings take a long time to grow. Infertile seeds are white in color, while fertile seed is black in color. The roots are thick and fibrous. Stevia is a short plant by nature, with a vital duration of approximately 13 hours. Stevia will begin flowering once it has developed a minimum of 4 true leaves. And stevia also uh, they have very low rate of germination. Malaysia still lacks suitable varieties and production technologies, while a few countries such as Japan and India have successfully produced suitable stevia varieties. Therefore, there is need to carry out appropriate research and development program to develop suitable local varieties. Mutation breeding is one of the strategies that can be used in the agriculture sector to improve stevia genetics and variety. When opposed to traditional breeding methods, mutation breeding has the potential to broaden diversity and isolate desirable economic traits in a shorter period. Among other physical mutagenic agents, the most frequently used mutagen in mutation breeding are gamma rays. So this is my research objective. Firstly is to identify the radiosensitivity of stevia mutation after being exposed to gamma radiation. The second is to determine the M1 generation morphological characteristic of stevia mutation after gamma exposure. Okay, now we proceed to the materials and method. For the plant material preparation, the plant material was prepared by the stem cutting. Stem cutting method is the most easiest and the most cheap. It's a very cost effective. This method is used in order to grow the roots from the stevia. The second is the seeds of stevia were irradiated with gamma rays to induce mutation plant. Okay. So we irradiate the seeds by using the BioBeam GM 8000 Germany machine at Agency Nuclear Malaysia. So by using this machine, we can produce a mutant seed for the third is you grow the seed, germinate the seed by using the red light. Method is prepared by the Abdul Latif et al. And this method is allows germination under lab environment. Next is we have to, after this is already germinate, we have to transfer the seedlings to the polybag and set up with the fertigation system. After that, the parameters were evaluated by qualitative analysis for the leaf. The quantitative analysis was measured on the plant height, leaf number, branch number, leaf width, and leaf length. Lastly, the data was analyzed by using SPSS analysis. For the results and discussion, the sensitivity of potential mutants is stevia. As we can see here, this is the table for the percentage of stevia mutation seed germination. Okay, we can see that the treatment seed from 100 gray record the highest rate of germination, meanwhile the lowest is from treatment 500 gray. So from the results above, we conclude that the rate of germination percentage decrease as the doses increase due to the negative effect from gamma radiation toward the stevia seed. Okay, so this is the survival rate of stevia seedlings. The survival rate is observed for 40 days after the seedlings is transferred to the polybag. Okay, so here we can see that the treatment for control recorded the highest, which is 75%. And the lowest one is from treatment 3 to 5 gray. And it is record 0% survival rate for both 400 gray and 500 gray. Okay, this figure shows that the survival rate percentage to gamma rays dosage is formulated by y equals to negative 0.1436x plus 76.192, where y equal to 50. Therefore, we can get the LD50 by using this formula. And the LD50 is 0.82.40, which means that the 50% of the stevia seedling with that is exposed more than 182.40 rate of gamma dose. Okay, we proceed to the morphological evaluation on gamma irradiation treatment. Okay, so here we can see that the qualitative morphological characteristic from this.
All right, good presentation actually, but due to the time constraint, I have to stop the ID number 91 presentations because it has been more than 7 minutes. So let's move to the next presentation.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Let me introduce myself. My name is Jatuh Charantika. I'm from Universitas 11 Maret, and my team is Professor Dr. Samahudi, SPMSI, and Dr. Insinyur Amalia Tetranisakia, MPMPhil. Our research is about organic materials and fish emulsion in shoot multiplication of Cavendish banana. There are two parts of my presentation today: introduction, material and methods, and result. The introduction. Banana is the fourth most important plant in the world, belonging to the Musaceae family. However, banana cultivation faces several obstacles, one of which is the availability of seeds. Banana monoculture cultivation in large areas encounter obstacle of the availability of healthy and uniform seeds. While the demand of banana is increased for the food consumption and industrial raw materials, banana cultivation on an industrial scale is constrained by the lack of availability of seed source. The effort to increase the availability of seeds can be made through tissue culture techniques. Tissue culture techniques are expected to produce seeds in bulk and in a relatively short time. The use of growth regulators in tissue cultures depends on the direction of growth of the desired. Combining primarily an appropriate growth regulator will increase cell division activity in morphogenesis and organogenesis. Material methods. The material is used. The material is using the modification of MS media that enrich with animal and vegetable organic ingredients according to treatment. There is certain treatment including MS media enriched by benzyl aminopurine as control. Data analysis using completely randomized design, one factor. Result and discussion. Level 1. Multiplication percentage. The result showed that the use of MS plus PAP and sweet potato extract plus fish emulsion for ppm showed the highest percentage of banana shoot multiplication, which was 100%. Untari and Puspitaning said that banana extract is known to contain the element of potassium, phosphorus, and iron, so it has a positive effect in shoot growth. And bean sprout extract, besides containing auxin, also contain the tryptophan amino acid, that the most crucial organic substance in auxin biosynthesis, and mineral such as calcium, iron, magnesium, phosphorus, and zinc, which play a role in shoot formation, said Saputri et al. Sweet potatoes contain several kinds of vitamins such as vitamin B, vitamin A, niacin, riboflavin, and thiamine, which function to accelerate the cell divisions, while the fish emulsions contain the element of nitrogen and potassium, and also contain the phosphor. Phosphor elements are much needed by plants for vegetative growth, which together with carbon, hydrogen, oxygen, and nitrogen elements, the phosphor will change proteins so that cell development can take place properly, said Salolo et al. And table 2, time emerge of shoots. Time emerge of shoot is one of the essential indicators in tissue cultures. And in this table, we can see that the media enriched with benzyl aminopurine still show the best result compared to the treatment using the organic methods at 3.74 days after replanting. This is because the benzyl aminopurine contains cytokinin that play a role in cell division, organ formation, and the formation of plant buds, said Villa et al. At 2011. The exogenous hormones added to the media will change the balance of plant growth regulator in cells. Exogenous growth regulators are given to provide a balance for endogenous hormones in order to be able to influence psychological response as a driver in cell division and elongation during shoot multiplication and shoot morphogenesis. said Kasuningachi et al. Table 3. The number of shoots. Here, still, the best result is
All right, actually a great presentation, of course, but due to the time constraint, I have to stop the ID number 138 because the presentation has more than 7 minutes. Okay, let's move to the next video.
Assalamualaikum warahmatullahi wabarakatuh. First of all, let me introduce myself. My name is Panji Harjanto and my partner are Imam Mujahidin Fahmid, Saleh M. Ali, and Aimal Bashir Demalino. We are from Hasanuddin University, Makassar, Indonesia. Ladies and gentlemen, on this occasion, I will present of paper number 104, title Institutional Development of Farmers to Agriculture Area-Based Corporation in Indonesia. Introduction. Organizing farmer. Organizing farmer has been the main strategy for improvement after in rural areas in Indonesia. 2. The problem is this group do not develop as expected, so they are not able to support of achievement of program goals. 3. Farmers Corporation are a very strategic form of farmer economic improvement because it will unite all the resources owned by farmers and manage them in integrated management. 4. The growth The purpose of the study is in the division of studying, also give recommendation for policy of the development and integration of farmer institution framework. The various activities carried out this world series of study are 1. Prepares study plans, extract primary and secondary data analysis and with problems. 2. Conducts on study and regulation policy and program for the development of farmer economic insertion. 3. Conducting analysis and completing research manuscripts. Research methods that tips and research. The synthesis of data and information from the literature review become the material for formulating policy recommendation from the study of development. Analyzing method. The analyzing method used is descriptive qualitative method. Data analysis. For certain case with it will be enriched with an in the depth analysis of that or information on the scope of the particular case of area. Data analysis. The study allocation cover major or strategic development program nationally. Development of farmer instruction and farmer economic institution national 2015 until 2021. Farmer corporations are built with a purpose. A. Increase farmer income and standard of living. B. Strengthen system. C. Increase added value and competitiveness. D. Increase farmer capacity and independent farmer business institution. E. Increase adoption of technology and agricultural mechanization that advanced and modern. F. Increase access for information technology in infrastructure, finishing, processing, and marketing. recommendation for policy for the development and integration for farmer institution and partner for the growth and development based to agriculture areas. The Ministry of Agriculture as pioneer cross-sectoral cooperation with other ministries or agencies and local government to optimize to potential carrying capacity. The implementation of various regional development partners with an area-based approach that has been developed by the Ministry of Agriculture. The step for the growth of farmer corporation and the integration of farmer institution in agriculture development areas will include several steps that must be carried out sequentially. At the preparation state for the growth of farmer corporation includes socialization of farmer corporation, consolidation of stakeholder, consolidation of farmer and farming business, design of farmer corporation, preparation of business models, determination of international from management and legal status, preparation of supporting infrastructure, readiness of human resource. Consultation and policy recommendation. Consolidation, 1. The Ministry of Agriculture is in the form of developing an approach to bridge to area-based agricultural development management by supporting the development of farmer institution. 2. The strength and the position of farmer. The government is also developing farmer economic institution. Policy recommendation. Agribusiness venture policy recommendation. 1. Agribusiness ventures in the modern world require strong actor. 2. Development of agriculture area development based on farmer corporation must be carried out with a regulator approach to strengthening farmer economic. 3. Institutional farmer in form of farmer group. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
All right.
Yeah, sorry, our inconvenience.
All right, so sorry. honorable participants in these rooms. I have low connections in my room here. So, okay, let's we move together to the chat box. There is some questions from Mr. Zulfahmi to the ID number 138. Mr. Jati Widnu. Can treatment number 0382 and 0182 treatment substitute of BIP for multiplication of banana? So please welcome Mr. Jati. Can you answer the questions?
Okay, thank you very much. Can the extract sweet potato and excuse me. Yes, can the treatment substitute the BIP for the multiplication? Yeah, in this data we can see that in other treatment, this treatment is the best option to substitute the BIP for multiplication of banana, because on time emerge and root, it shown the best excuse me. It's shown the best result on this treatment. Yeah, for the change of this treatment can be used to substitute the BIP material. That's thank you.
Okay, thank you very much, Mr. Jati Widnu, for the great explanation. Or maybe there is some feedback for from Mr. Zulfahmi to Mr. ID number 89. Is there any feedback? Okay. Okay, nothing is enough clear. Right. So, ladies and gentlemen, now we will move to the last sections of our session in this parallel group. Yeah. There will be 3 last presenters in this room. Wait a minute. All right. There will be presenter with the number ID 140. I will say hello to Mrs. Sydney. Is that right, Mrs.?
Yes, I'm here. Yes.
All right, Mrs. Sydney, thank you for the confirmations. And then the second speaker, there will be with the ID number 146, Mrs. Elinor. Miss Elinor, or all right.
Yeah. Yes, I'm.
All right. Yeah. Thank you, Miss Elinor. And then the last presenter will be with the ID number 166, Miss Sugi Harti.
Yes, yes, I'm here. Fanny.
All right. Okay, Mr. Fanny or Mrs. Fanny, I should call.
Mr.
All right, Mr. Fanny. Thank you very much for the confirmation. So,
Mr. Fanny, Mrs. Fanny.
All right, Mrs. Fanny. Okay, so thank you very much for the confirmation. Now, ladies and gentlemen, let's we watch together the video presentation for our best presenters. Time is yours for the organizing committee.
Video katanya, Bu? Iya, nanti pertanyaan. Hah?
For giving me the opportunity to explain my presentation. On this occasion, I would like to deliver my presentation with the title The Diversity of Wild Tampoy, Bacoria Pilantacea, and their Potential for Improving Livelihoods for Local People in Aceh, Indonesia. My name is Zine Oman Navia. and with our team from Universitas Samudra Langsa Aceh, Indonesia. We know that Indonesia is known as the center of the distribution of tropical fruit plants in the world, with more than 80% of them found growing wild in tropical forests. Local communities, including in the province of Aceh, have used various types of these plants as a source of food and medicine, one of which is from the Bacorea groups. However, there are still many wild Bacorea species in Aceh province whose potential has not been fully explored. In addition, several types of wild Bacorea are still unknown to science, so new species or varieties have the potential to be found. The increase in population has resulted in an increase in the rate of deforestation, which has an impact on the decline in the Bacorea population and has the potential to cause the extinction of the species before its potential is explored and utilized optimally. The study is essential to do as an effort to conserve the gem plants, primarily the wild Bacorea indigenous to Aceh, for the purpose of sustainable use and to support the improvement of nutritional quality and public health. The study was conducted from June to July 2022 at South Aceh and Southwest Aceh District, Indonesia. The map in Figure 1. The plant specimens were collected with an exploratory method, and information on the use of Bacorea was gathered from literature studies and interviews with local people. Based on the result of the study, the diversity of Bacorea species in the research location was found to be 8 species. From the table in the site, it is known that in South Aceh there are 8 species, while in Southwest Aceh there are only 6 species. Here are some pictures of Bacorea plants found in the study site. This is Bacorea lanceolata and Bacorea brevis. This is Bacorea sumatrana and Bacorea polyneura. And the last, Bacorea parviflora. The potential of Bacorea at the study site is utilized by the community as a source of food, medicinal raw materials, and building materials or construction. Based on the table on the slide, it shows that all of Bacorea in the study set can be eaten, while for medicine there are 5 species, and as construction materials there are 7 out of 8 species. The findings of this study show the importance of Bacorea role in supporting household livelihoods in different ways, including food supplies, medicines, and constructions. For the last, we are grateful to all people in the study village for their kind hospitality and share of knowledge. The authors thanks Indonesian Ministry of Education, Culture, Research and Technology, Kemendikbudristek for funding support with PDKN grant. That's our presentation. I would like to say thank you very much.
Assalamualaikum warahmatullahi wabarakatuh. My name is Elinur from Agribusiness Department of Universitas Islam Riau. My member is Asrol Herianto Siska Paulina. I want presented my our paper title Profit Function Catfish Farming for Increased Household Income in Pekanbaru City, Riau Province. Research background. Catfish, one of its freshwater fish that is widely cultivated by fish farmers in Pekanbaru City because catfish harbor age is very short. Grow fast, maintenance is easy and profitable business. Catfish, catfish is consumed by households in Pekanbaru City because it is delicious, contain amino acid, leucine and leucine needed by the human body. Catfish can substitute of other fish for household consumption in Pekanbaru City, where the availability of seafood depends on the season, but catfish is always available at any time. We can see catfish production in Pekanbaru City 2018 and until 2020. The figure is the figure can see the average growth of catfish is 0.16 ton per year. The increasing production indicate fish farmers are interested in cultivating catfish. Catfish farming can continue depending on profit of catfish farming. Catfish farm make decision in allocating a production factor to achieve maximum profit. Input for catfish farming are ponds area, labor, seed, and feed. In the theory, profit function depend on output and variable input price. If output price increase, the profit will increase. But if the input price increase, then profit will decrease. Result of review study, farming profit are determined by price of output, variable input price, and fixed cost. Thus, the concept of profit can be applied to the catfish farming. Research objective number 1, to analyze profit level of catfish farming. Number 2, to analyze determinant at catfish farming profit. And the 3, to analyze response of output and input price to catfish farming profit. Research method is a survey and location in Pekanbaru City, Riau Province. Population was all catfish farmers in Pekanbaru City. Samples were taken for 4 districts, for example, Tenayan Raya, Tampan, Bukit Raya, and Rumbai Pesisir sub-districts. Sampling method uses simple random sampling, a number of samples of 98 catfish farming. Data collection model was interviewing catfish farmer with questionnaire. Method of analysis, the first farming analysis. The second, inferential statistical analysis with multiple regression. The profit function is in the slides. The second of method analysis with elasticity of output and input price. Result and discussion. The first result, profit of catfish farming in Pekanbaru City. The table consists of fixed cost, variable cost. Fixed cost consists of depreciation. Variable cost consists of wages, seed cost, or feed cost. The large production costs to catfish farming in Pekanbaru City was feed cost was 78.02%. Catfish farming in Pekanbaru City profitable. The second result Determinant of catfish farming profit. The table we can see a price of catfish, wages, price of feed PF 1,000, and depreciation are significantly effect to catfish from profit. But price of seed and price of feed PF 800 is not significantly. Overall, this model statistically good. We can see R square and F6 probability. The third result was responsive of catfish price and input price to catfish farm profit. The table can see price of catfish responsive to catfish farm profit, but wages, price of seed, price of feed, and depreciation is not responsive to catfish farm profit. Its mean change in catfish price have a big impact on change in profit farm. In other words, increase in catfish price will be able to eliminate an increase of feed price. Thus, catfish price policy is needed. Conclusion. Number 1. Catfish farm in Pekanbaru City is profitable and feasible. The production cost of the catfish farm consists of depreciation cost for fund and equipment, labor cost, seed, and feed. Cost of feed is the largest contribution to profit cost of catfish farm. Number 2, significantly factor effect to profit catfish farm are price of catfish, labor wages, feed PF 100 and disposition cost. Meanwhile, price of seed variable and feed PF 800 variable did not significantly affect to profit farm. Price of catfish.
All right, actually a great presentation that talked about catfish, but due to the time constraint, I have to stop the ID number 146 because the presentation has been more than 7 minutes. So we move to the next video.
My
I'm sorry,
article with
Mr.
the
Raj.
entitled Value Chain Mapping of Porang Commodity or Amorpholus muelleri in Wonogiri Regency. We are from Agribusiness Department, Faculty of Agriculture, University of 11 Maret. I'm Fani Widadi. Introduction. As we know, porang or Amorpholus mueller is a commodity that has high value economic, mainly for export. According to Indonesian statistic data, from January to September 2020, the export value of porang is until 331,000 US dollar, or is nearly 90% of porang production in Indonesia is for the export market to China and Japan. As we know, porang is used as raw material for several industries, such as paper, pen textile, and glue. In addition, porang has a high content of glucomonans, so it can also be used as ingredient for electrical insulation, toilet paper, and also cosmetic. Porang can be processed to be flour and is source of dietary fiber. So it's used also for food such as noodles, tofu, and some Japanese foods such as konnyaku, shirataki noodles, cocktail ingredients. According to our survey in Wonogiri in 2021, the price of porang taber at the farmer level is around 3,000 until 3,500 per kilo. And then if the tuber has been processed to be dry or in here to be chips, the price will slightly increase until 70,000 until 22,000 per kilo. And if it will be processed further to be into glucose and flavor, the price rose until 125,000 until 150,000 a kilo. Even though the economic value of porang is high, there are several problems of value chains in Wonogiri still exist. The first is the price of porang, value added, and also market share at the farmers level chains is lower than the other actor chains. And then also the asymmetric information appears that is between the farmer and wholesaler. In here, farmer don't have many information about the price than the wholesaler. And also the low coordination at the upstream chain level between the farmer and buyers. Therefore, the purpose of the study is one to describe the mapping value chain of porang in Wonogiri Regency. We will, in this paper, we will describe the value chain mapping, including the flow of the product, information, and also financial. In addition, we also explain the role of each actor is to increase the value of porang. For the collecting data, for the primary data, we get the data through interview all the actor the chains, such as farmer, wholesaler, at the rural level, at the sub-rural level, and also big wholesaler that have the contract to supply the porang to the processor. The respondent we select through snowball sampling, and here the number or the more number of the actors that we interview to get the data of the value chain mapping. And for the secondary data, we get data through the report from the Agriculture and Food Services at the Wonogiri Regency, Wonogiri Regency Central Statistic, and also Agriculture Extension Center. For the analysis, we use value chain mapping analysis. The first thing is we identify actor who play role in marketing porang from the production until the final process, and also we identification the flow of information, financial, and also product from the upstream to the downstream value chain. This is the production, porang production in Wonogiri. We get the data from the Agriculture and Food Service at the Wonogiri Regency at the 2021 that the total land area in Wonogiri is 142,000 hectares, and the highest number land area is in the Karangtengah district. This is our result of the paper. This is the value chain mapping. We found 4 value chain mapping in the Wonogiri Regency. The first is value chain number 1. This is the farmer sell to directly to the processor. And then the value chain 2, porang farmer sell to the wholesaler at the sub-rural or district. And then the sub-rural sell to the big wholesaler goes to the processor. And then the value chain 3, the farmer sells to the big wholesaler, and then big wholesaler goes to processor. And then the longest one is the value chain 4. This is for farmer sells to the wholesaler at the rural level, at the sub-rural level, big wholesaler, and then goes to the processor. And in here, every actor in the chains, there is the flow of the product, financial, and also information. The information flow such as quantity, quality, and also agreement between the actors. This is the role of each actor in the chain. The first thing, farmer porang have main activity about the cultivation activity. They do land preparation, seeding, planting, maintenance, harvesting, and cleaning. And then the wholesaler in here at the rural level, super rural level, and big wholesaler have main activity to distribute and also marketing the porang until to the processor. In here, for instance, at the wholesaler at the rural level, they purchase of porang at the farmers, and then they do marketing and also sorting. And the last actor is the processor. They have main activity to process the porang tubers to be the porang flour. In here, the processor doing something purchase of porang from wholesaler, and then they sorting, washing, and then drying, and also processing into flour. This is the mapping of value chains. Mapping of value chain in the monogiri regency. That the farmer focus on the production activity, and the wholesaler focus on the distribution, and then the processor is focused on the processing that process porang tubers to be porang flour. The conclusion of this study is the first. There are actors involved in the value chain of porang. There are farmers, wholesaler at the rural level, at the sub-rural level, and also big wholesaler that have the contract to supply porang, and also the processor. We found for mapping of value chain of porang regency, and every actors in here has activity to flow of the product, information, and also financial. The farmer in here focus on the production, and the wholesaler focus on the distribution activity, and the processor focus on the processing of porang. This is our presentation. Thank you for your attention. If any questions, let me know. Thank you.
Thank you, Mr. Fani. That's great presentation actually from our three presentators, of course. And now we move to the discussion times. Here's in the chat box, there is some questions from Mr. Sulfahmi again. Wow, it's very amazing. Mr. Sulfahmi have a critical thinking, of course. So he always asks something to these presenters. Mr. Sulfahmi asked to the ID number 140. To Mr. Sydney and Tim's, to Mrs. Sydney and Tim's, the question is, Bicorea has been used for medicinal by local people. It is very interesting. Can you explain more detail? What is it for? Okay, Mrs. Sydney, please.
Yes, thank you for your question, Mr. Zulfahmi. From our research, from the public information, that Bacodia lanceolata can use as a stomach ache medicine. The ferric cup of fruit is boiled, then the water is drunk for use stomach ache. Well, the study literature that Bacodia brevetibus can use menstruation regulator, and Bacodia polyneura rich in vitamin C and antioxidant. Thank you.
Okay, thank you, Mrs. Sydney. Is there any feedback from Mr. Sulfahmi, or is it clear enough for the answers? All right.
It is very clear.
All right. Thank you.
Thank you, Mr.
Yep. So, ladies and gentlemen, good job all of the presenter here giving such an informative and comprehensive presentation from the first section until the last sections. It's really increasing our knowledge to know about the agriculture productions, biotechnology, and economics, of course. We are now on the last sections. Now, I would like to ask all of the participants here to turn on the camera and we will have photo session together. Baik Bapak Ibu semuanya, dengan hormat saya memohon untuk Bapak Ibu bisa mengaktifkan kamera. Kita akan foto bersama-sama. Saya akan pandu dari sini dan I will ask help for the operator organizing committee, Mr. Gali, to take a photo. Okay, I will count the numbers. 3, 2, 1. Cheers. Okay, once again. 3, 2, 1. Cheers. All right, thank you very much, all participants here. There are some information that I have to inform to you, all of you. The A certificate for presenter will be delivered within 3 days by the committee via email, and then the information regarding to the publication and payment of the publications will be announced later. I should remind to everyone here that we will have General Assembly on 25 August 2022. It said that tomorrow via Zoom meeting or YouTube, the link had been shared by the committee or every people can view it in our website. I would like to close this forum by thanking to all of the presenters for their participants in our parallel session of the 9th International Conference on Sustainable Agriculture and Environment, Universitas 11 Maret Surakarta 2022. Finally, let's say Alhamdulillah together. Alhamdulillahirabbil alamin. I say thank you very much for the attention and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Warahmatullahi wabarakatuh. Nongol sekarang. Siska Palina. Oke, alhamdulillah.
Alhamdulillah, thank you, Ms. Elinor. Thank you, everyone here, Mrs. Sydney.
Okay.
Thank you, everyone.
You're welcome, Ms. Aida, Mr. Panji.
Thank you.
Okay, we will leave these rooms and we want to end this meeting. Thank you very much, everyone.